You look so serious, Laura.
You look so tropical.
I know. I figured with the like 10 feet of snow outside, this was a fitting background. It looked, I went out and fed my horses just a moment ago, and my border collie dog's out there like all excited just to be looking at animals. And it looked like he's laying down and he's like standing full in the snow, but he just like his body's barely poking out of the top of the snow.
I saw a picture from Sheva of her house, and she looks like she's buried.
Oh, well, these drifts are amazing. Like, ugh, my front door is inaccessible, but
Really?
yeah. Yeah, there's like, I don't know, 3-4 foot drift outside my front porch there.
Wow.
But yeah, I'm kind of thinking because next week are these scoping meetings, and I'm like, I don't know if I want to drive to Elko.
Oh, right. Is it supposed to let up or supposed to keep going?
Well, I mean, I don't know, I'll have to look again. They keep updating it, but it's supposed to be on and off for a while more, I think.
Well, it'll be good for the water situation.
Yeah, for sure. I'm excited about the conversations today.
Yeah. It's always the case, so that there's more to discuss than we have time for. So it'll just be a start on some of this stuff.
Yeah. So are we just waiting for more people to join, or?
Yeah, 8:15 is the start time.
Oh, silly me. I'm gonna go
Look, you're
refill.
early.
Crap. Well, I'm always either early or late. Now you know the secret. Just make me think it starts earlier.
I know. Sharmila, can you hear me? Nobody's talking. That's probably why I can't hear anything. Okay. Hi, Melanie. How are you?
I'm over shoveling snow. How about you?
It sounds like you all are. I got to take a trip to the San Francisco airport and hang out there for a little bit.
Lucky you. I'm sorry.
Yeah, and then fly back. It was great.
Oh boy, that's what you wanted to do, was more flying around, huh?
Yeah.
Or are you about ready for us to get things kicked off here?
I'm ready. I'm jumping on Teams here because somebody just started the meeting.
I'm gonna announce in the room to get people circled up and then grab Rachel.
Yep. And then let me know if you want to do a round of introductions or who's all in the room.
It's all the same people as yesterday.
Okay.
Yeah.
Faith, can you do me a favor and pop the Zoom info into the Teams link? Because there's someone on there.
Into the Teams link?
Yeah. Did I say Rachel? I meant Faith. Sorry, I was looking at you.
Okay.
Yeah, some people started the Teams meeting and I don't want to pull too many things up on my computer because it may crash it.
Did you hear? She said, can you put the Zoom link in the Teams chat because someone keeps joining there? Yeah.
Thank you.
And then how do I start recording?
It's already
Oh, okay.
recorded and I'm recording too, so we're set.
Okay. Well, that's okay. She was talking to me. Well, when she's gonna talk, I'll put it down. All right, welcome to day 2. I'm surprised that so many of you have returned for today. Okay, so I'm Rachel Van Horn. I'm the Forest Fish Bio for the Humboldt Toy Abbey. And just so y'all know, Sean had to take Yashi to the hospital yesterday and they are there now. So he will not be joining us today, just so you all know. He said that we should let you know that things are okay and they're just doing some tests. All right, so to recap from yesterday, we are carrying on today with our genetics conversation. And so yesterday we heard about we still have one topic. Coulter's going to go next, but we need to work on this development of the genetics management plan. It's one of the things in the 2019 UGOs that we really haven't started yet. And so the purpose of the genetics presentations that we heard yesterday and then Coulter's today is to introduce these topics to you, for you to sit there and chew on this information, and then to identify challenges and potential solutions. And so yesterday we were supposed to have like a 2.5-hour time block to go through those things, and we didn't. And so today we've kind of cut it down to 90 minutes where we split it out into kind of the topics of those presentations. We're going to have 20 minutes for each topic to kind of have people have their questions that they want the genetic management team to address, potential solutions, and insights that they feel like once that team is formed that they need to bring forward. All right, so we're hoping to get the genetics core team put together in this spring, April-ish. So if you have interest in being a part of that, reach out to Rachel Humans about your wanting to participate on that core team. And once that gets established, she'll make sure that emails go out. Okay, so the purpose of this genetics workshop today, what we're going to do that 90 minutes after Coulter presents, is to take a slightly deeper dive into the challenges and potential options related to the topics that were discussed in the presentations. So we understand that there's probably going to be a lot more discussion than we can have in a 20-minute time frame per discussion. So if your questions don't get answered, make sure you write them down and send them to Rachel or Sean or Faith so that they can get added to that giant list that the genetics management team can work with. So our first topic that we're going to talk about during that 90 minutes is the genetic rescue / isolation topic, and then talking about drift versus selection, talking about Fish and Wildlife Service's 5-year review criteria for hybridization versus what Ryan kind of talked about yesterday and how Montana deals with hybridization in their hybridization plan. And then evolutionary significant units. So what Robin discussed yesterday about, do we go as far as DPS is RIT already our kind of definition of that? That sort of is one of the topics. And then basically a strain discussion. What strain goes where and why? That is the last one.
And
All right.
Rachel,
And
can I
we
just
can-
pop in for 1 second? I think we're gonna table the 5-year criteria, the Ryan's criteria and the 5-year review criteria, because Ryan is gonna be with us today. So we may need to we'll need to follow up on that in more detail at a later time.
Laura said that we'll discuss the hybridization at a different time because Ryan isn't going to be here to field questions. I have to find out where your mic is, Cody, so I can swing it down. All right. So with that up, Coulter is going to talk about reintroductions as a whole. So vacant stream, putting fish back in. Do you see the presentation, Laura?
Yes, go down to the bottom and click on the presentation view.
And then
And then display settings up top. And duplicate slideshow. Yep.
It's almost like I watched Sean do it yesterday.
Rachel, how do I
Oh.
get that there? All right, so I promise this is going to be the last genetics talk. We'll quit shoving genetics down your throat after this. But because we recognize that reintroductions are probably going to be a necessary conservation tool to restore the hot and cutthroat trout across the landscape, we thought that it would be beneficial to have a discussion about how Montana Fish, Wildlife and Parks has approached reintroductions. Okay. And the resulting outcomes from what they've done recently within the last decade.
Hey Coulter, is it possible to adjust that computer just a little bit so we can see your face instead of your chin? There
How's
you
that?
go. Thank you.
The arrow thing isn't working here.
Yeah, he clicked off your presentation.
Oh.
Yeah.
Oh, there we go. Okay.
Uh-oh, the room just froze.
Same for me too.
Technical difficulties. Let me text someone over there.
All Westfield cutthroat trout populations are represented by these colors, either red being at risk, yellow being considered protected, and green being considered secured. And you'll notice that on this map, a lot of these populations are considered at risk, and this is for two reasons. One being that they're at risk of hybridization with rainbow trout or other competition with non-native species. And the other being that many of these populations are small, isolated, and often lack genetic variation. And as we kind of discussed yesterday, this is typically concerning because small populations are often highly susceptible to strong genetic drift and inbreeding depression, which can in turn affect the fitness of these populations and potentially reduce their adaptive potential to future changing environmental conditions. And so this is where reintroductions come into place as a potential conservation strategy where we reintroduce populations back across the landscape. However, there are still a lot of questions regarding the efficacy revolving around reintroductions and the outcomes from those management actions. And so really, as conservation managers, we're left with kind of this overarching question, whether it be Westfield cutthroat trout or any other subspecies of cutthroat trout, and that is, how should we go about reintroducing fish back into the landscape? And so commonly when we think about reintroductions, we often have a system where cutthroat trout were once present, but due to invasion and introductions of non-native species, cutthroat trout no longer exist in those systems anymore. And so to recover cutthroat trout back into these systems, we often have to remove non-native species above either artificial or natural stream barriers and then reintroduce cutthroat trout back into that system. And one of the larger questions involved in how do we go about reintroducing fish is whether or not we use multiple source populations or a single source populations to refound that new population. And both single and multiple source reintroductions have their potential benefits and consequences. For example, single source population reintroductions offer the opportunity to replicate a single population on the landscape. However, given that many of these small or remaining cutthroat trout populations are often small and isolated and lack genetic variation, it often begs the question, can these populations adapt not only new conditions, but changing environmental conditions? And on the other hand, multiple source reintroductions offer the opportunity to boost genetic variation if population mixing occurs, as well as preserve the genetic legacy of or any important genetic variation of several populations. However, this conservation strategy does come with its assumptions, several actually. The first being that genetic variation is correlated with fitness, that hybridization between these populations is actually beneficial, and that we would expect hybrid vigor or heterosis, and that outbreeding depression, which we talked about a little bit yesterday, which is essentially the decrease of fitness in hybrids, does not occur. And really, it's decisions like this and these types of strategies that ultimately govern the abundance, genetic variation, and the viability of these reintroduced populations. And so we also talked a lot yesterday about the importance of genetic variation in conservation genetics, and it's often deemed as the gold standard in how we go about conserving populations. However, its importance in conservation genetics is still somewhat debated amongst researchers, and this is primarily due to the lack of empirical data linking genetic variation to population persistence or viability within a natural setting. And so actually, not only can we address how to reintroduce, or the best practices for reintroducing cutthroat trout into a new system, but we can also use the practice of reintroducing multiple source populations to evaluate exactly how important genetic variation is to these populations. And so, like I mentioned earlier, there are often many questions involved in how we go about reintroducing fish and the resulting outcomes from those management practices. Several shown here. For example, does the genetic variation of source populations matter or influence how successful populations are following reintroductions? Again, is hybridization beneficial? How quickly do these populations rebound after being reintroduced, et cetera? So there are often, like I mentioned earlier, a lot of questions involved in reintroductions that we just currently do not have many answers to. And so to begin addressing some of these questions, we actually evaluated 4 West Slope cutthroat trout reintroductions in Southwest Montana, which is shown in this map to the right here, with reintroduction streams shown as stars and circles representing source streams. And then the colors correspond to where fish were translocated to within Southwest Montana. And then I also want to point out here in the upper right-hand corner of this map, we show estimates of genetic variation of these reintroduced populations. And you'll notice that genetic variation varies pretty widely among source populations that were reintroduced, which could in turn have an effect on how these populations or the success of these populations following reintroductions. And then another thing to mention here is that within all of these reintroductions, these were performed using wild fish translocation. So there was no hatchery involvement with any of these 4 reintroductions. So a little bit of background here into how fish were translocated. They were moved between 2015 and 2018, and source populations were sourced mostly equally among populations, with the one outlier being the Ruby reintroduction system, where only 10 fish were actually sourced from one of the source populations. And this was due to a lack of, or that source population being extremely small and the ability to take more fish from that system. And so to address the questions that I mentioned earlier, we collected genetic samples from first genetic offspring in 2019, or from first generation offspring in 2019 and 2020 from all of these 4 reintroduction streams, and then completed a series of population contribution analyses, assigning these first generation offspring back to the population of origin, or the source population from which they came from, to again address these questions. And just quickly to make sure that it makes sense on what we did here, if 2 fish from population BN produced or reproduced, we would call that, we would assign that offspring a cross type BN, calling it a parental cross type because it only originated from the one population. Whereas if a source population BN and PR reproduce, we would call that a hybrid, assigning that a cross type such as BN-PR. And then one of the other questions that we wanted to address with these reintroductions is how quickly these populations rebounded following reintroductions. Essentially, and to do so, we simply evaluated compared the population size of non-native species prior to reintroducing West Slope cutthroat trout to population size following reintroductions. And this was mostly just to gauge really how quickly these populations were able to fill habitat following a reintroduction of a given number of fish in that system. So to start here with some of our results in this is population NFG with the 3 source populations that were translocated. We saw an 87% increase in West Slope cutthroat trout abundance compared to pretreatment non-native abundances. And then also observed a 46% increase in genetic variation compared to the mean of the 3 source populations that were reintroduced. And then here getting into our population contribution results, which is shown in this graph to the right here, with the number of offspring on the y-axis and cross type on the x-axis. And starting with parental cross types with the number of observed progeny that we sampled and what we would have expected if these populations all mated randomly and there wasn't some driving force affecting how population or how successful some populations were. And so the first thing to point out here is that the source population with the highest genetic variation had nearly 3 times as many offspring as we would have expected. Whereas the cross type with the least genetic variation, BC, had 5 times less offspring than we would have expected. And then in looking at hybrid cross types here, the first thing to point out is that the hybrid cross type between the 2 most genetically variable populations had more offspring than we would have expected, whereas both cross types with BC ancestry had less offspring than we would have expected, suggesting a potential negative consequence of having BC ancestry or that lower genetic variation. Transitioning to SFG results, we again saw a high increase in the number or the abundance of West Slope cutthroat trout compared to pretreatment non-native abundances. And then also again saw an increase in genetic variation compared to the mean of the 2 source populations. And similarly here with population contributions, we saw that the cross type with the least genetic variation, CW, did not have as many offspring as we would have expected. And that the hybrid cross type had more offspring than we would have expected, potentially suggesting a benefit of being a hybrid in this system. In Pete, we saw somewhat different results where we saw a 76% decrease in the number of West Slope cutthroat trout compared to non-native abundances. And we believe that this is likely linked to the number of fish that were translocated for the amount of habitat that they were translocated into. We don't think that this population is necessarily going to continue to decline. It was just, we evaluated it, almost immediately following reintroductions, and we're chalking this up to more of like a slow expansion situation. However, we did see a 25% increase still in genetic variation. And in population, looking at our population contribution results here, we saw that both parental source cross types were underrepresented in the system, whereas again, hybrids were nearly 2 times. There were nearly 2 times as many hybrids in this system than we would have expected. And then in Ruby, we again saw a decline in the abundance of West Slope cutthroat trout compared to non-native abundances. However, did still observe an increase in genetic variation. And then looking at contribution results here again, we saw that both parental cross types were underrepresented, and again, offspring from the hybrid cross type were overrepresented, again suggesting a benefit here of hybridization. So to summarize and kind of put together all these population results, we wanted to evaluate the relationship between the success of these cross types and genetic variation, or in this graph, observed heterozygosity. And we found that genetic variation and hybridization strongly predicted reintroduction success overall in these 4 reintroduction systems, with hybrid cross types having both increased success and higher genetic variation. So then to take this analysis a little bit further and to confirm our population level results and examine drivers of reintroduction success, we then took first generation offspring in reintroductions NFG and SFG and assigned offspring back to their parents to evaluate fitness metrics such as family size, survival and reproduction, and reproductive success. And so just jumping straight into our results here, and starting with family size. With full sibling family size plotted on the y-axis and cross type on the x-axis. We first saw that cross types with higher genetic variation, BN and PR, had significantly higher family sizes relative to the cross type with the least genetically variable cross type BC. And then saw that the hybrid cross type between the two most genetically variable populations, BN and PR, also had increased family size relative to the other hybrid cross types with BC ancestry. And then I also wanted to point out here, each family is represented by either a blue or a red or pink dot in this graph. And you'll see that overall, hybrid cross types had more families relative to parental cross types, again, suggesting the benefit of hybridization here in this instance. And so then jumping into our survival and reproductive success results. This is actually really interesting. We ended up observing a strong positive relationship between both fitness metrics. So the probability that an individual would survive and reproduce and reproductive success, and that relationship to individual heterozygosity, suggesting that at an individual level, individual heterozygosity was highly influential of whether or not these essentially individuals survived to reproduce and then reproduced. And then in the South Fork, we did a similar analysis, evaluating family size first here. We did not see a relationship between cross type, genetic variation, or family size. However, we did see, similar to NFG results, that more families were produced from hybrid cross types. And then jumping to our survival and reproductive success results. We also observed a positive relationship between fitness and individual heterozygosity. However, this relationship was a little bit weaker compared to NFG. And for the sake of time, I can address that in some questions later. But to summarize all of these results and kind of wrap all this up, so a couple points to hit on here is that direct translocations of wild fish worked in all instances, increasing genetic variation, and that when we're reintroducing fish into small, or I should just say not small, headwater systems, hatcheries may not be a necessary component if we have source populations to draw from. We also saw that habitats filled faster when more fish were translocated. That genetic variation did increase in all 4 of these reintroductions. That populations with lower relative genetic variation were less successful. That population mixing increased cross-type success, so hybridization was beneficial in this case. And forgot to mention, I'm sure everyone picked up on it, but I am talking about good hybridization here, not hybridization with rainbow trout. In NFG, then we found that individuals with lower genetic variation had reduced mating success, and we saw a similar relationship in SFG. However, the relationship was a little more convoluted compared to NFG. And I will take any questions.
How long had these populations? How long have these populations been isolated from one another?
That's a good question. We don't know the exact time there, but we're assuming it's somewhere between 200 and 500 years, most likely. But I will note that these populations, when evaluating genetic differentiation between these populations, I have a graph or a deal here to pull up. So FST is just an estimate there of the genetic differences between these populations. And so we don't think that they've been isolated for super long, but in some of these instances, like with the Ruby reintroduction here, FST values were pretty dang high, but among these two populations. Yeah.
This is Rachel Van Horn. So in the reintroductions where the heterozygosity was really low, were the genes that were left in those populations different than the ones that had higher? Meaning, I mean, I think the answer is no, because once you cross them, it didn't necessarily, it didn't make more than the high one had to begin with, but because in a lot of our situations, we have one population left within a basin that is like the original, and everything else is kind of mixed, and people treat that as more important because it is the only remaining original stream we have left, but it may be like the lowest heterozygosity, right? And so what your thing is saying is that potentially in the long term for fitness doesn't matter.
The lower genetic variation of isolated populations.
Even though it's our only remaining original strain for that river system.
Yeah, so I think the biggest thing to take away from this is that, so yeah, like we saw a strong relationship between genetic variation and fitness, suggesting that, you know, populations and or individuals with more genetic variation were more fit than populations with less genetic variation. But I don't think that should take away from the importance of then preserving the potentially unique genetic variation in these populations that have low genetic variation, because it's not the same, you know, the genetic variation found in population X on this mountain range and Y on this population isn't the same. And there could be useful, you know, alleles within each of those populations that when you then combine them, it gives them a benefit of being hybrids in these systems, which is typically what we saw in that combining these populations together was beneficial for some reason, whether that be that it gave them the ability to, you know, essentially adjust to these different habitats or whatnot, or because that was an increase in genetic variation. Hopefully that somewhat.
I think so.
Okay.
There's a comment from Mandy here about these analyses would be great to do with Paiute cutthroat trout, considering recent translocations. They've started, but they don't have the sample sizes.
Yeah, and I don't know how to.
Or Mandy, sorry.
Hey, Laura, hang on just 1 second. We're gonna get a comment from Andrew in the room, and then I just want to kind of reframe, like, we're gonna enter into this whole genetics discussion here. We've only got 90 minutes to discuss all of the stuff from yesterday. And so I want everybody, Coulter, stay up here because I think what he presented was incredibly valuable to what we're trying to do. But I want to kind of open it up to talk about LCT in general, to frame this in the genetic rescue conversation, because we've only got about 20 minutes for each topic. And so let's try to keep it on genetic rescue, and then we'll kind of keep progressing. And then Andrew, go ahead with your comment, and then I'll address some of the Zoom comments.
Just real quick to add on to what Coulter did not answer, I think it's important to consider the relative variation. So BC had low variation compared to BN, performs poorly, but if it had been competed against, say, CW, it might have done as well or better. So kind of who you're in there with is important for that.
Do you just want to?
Coulter, can you repeat what was just said for the question? We didn't hear it on Zoom.
Yeah, do you need both the question or well, so Andrew just chimed in and said that it's important to recognize the relative genetic variation of these populations. So like an instance of population BC in reintroduction stream NFG, that population may have done poorly in NFG because that population had low genetic variation. But if you pair that population BC with a population like CW, who then has even lower genetic variation, the outcome might be different for BC.
Right, let's figure out this mic
Right.
situation. Right, Laura, we need to figure out the mic situation really quick.
Okay.
Mixing fish is very similar to
In the meantime, we'll do another.
You just want to. Okay, we'll just roll, okay.
All right, can you talk, Laura?
Yep.
Okay.
Can you hear me? Everybody in the room?
Okay, perfect, yeah. That's fine. I've never given
Like,
that. Oh, yeah, no.
let's
I
mute
didn't.
this.
There's not a way to Bluetooth to a speaker like we did at Folk Life Center, is there, Cody?
Oh, it's good.
Try that, Laura.
Can you hear me?
I wanted to hear it.
I can keep talking. I hear you guys have a lot of snow over there.
That's like when I got.
I don't have any jokes lined up.
Quick audio setting.
Right around like 3 minutes.
Yeah, you can at least bump her up a little bit.
Oh, no, yeah. Important to keep things rolling, otherwise we get in that spotlight like yesterday. Yeah.
All right,
Talking?
go ahead.
Can you hear me better now?
Yeah, but we need to figure out where,
I mean, we can have these conversations.
all right, go ahead and talk now.
Whoa, that microphone is coming right at me.
No, I know, we're trying to find out where the
No, I know.
microphone is on his.
I'm just laughing how it just came. I have to talk about something.
Yes.
I sound muffled. Okay,
Well, when did-
Tina's saying that, but
Just try that, Laura.
Tina must be online, so I guess I sound muffled online. I might sound nasally because I think I'm getting sick.
That's why I was wondering if it's worth asking.
It's a fan. Yeah.
We all like, part of the reason why we, you know, mix it.
Has something changed since yesterday or was it bad yesterday too?
No, this is, we had Sean's computer yesterday.
Oh,
This is
right.
Cody's computer, so we're just trying to.
Yeah. Can
You
we-
went to the mic, right? The mute setting and the up caret, and you can get on, test your speakers and your microphone to make sure they're both cranked up as far as they can go.
How about we just have Jason run the room here?
Okay,
I can keep track of what's popping up in the chat here and we'll relay that message through here on to the room on it. We'll
Okay.
just go with
does that sound better? I just increased my
Yes.
That was
microphone.
so much
Oh,
better.
that helps
Oh.
tremendously.
Okay, I just changed it. I guess yesterday it was set higher. So everybody that's on Zoom, if you're gonna have a question, if you would just take 2 minutes to go to the mute button on the lower left and click on the up caret. And then at the bottom of that list, you'll see audio settings. Open that up. Cody, you do the same on your end and make sure your speaker and your microphone is all the way up to the full,
We're all-
volume. Hopefully that'll help. Hybrid meetings are always a challenge.
See, 'cause I think the speaker is.
That's a fan. I could feel the air blowing out of it. Give us one more time, Laura.
Okay, can you hear me better now? Am I loud?
All right, one more time.
Okay, I'll keep talking about nothing.
Okay, so
Okay,
we're
there-
getting closer to it. Sorry.
Allie, Kelsey, can you?
Can you hear me? Loud and clear in the back?
Yeah, they're saying it's good enough. Well,
Okay,
we're gonna roll with it.
perfect.
Okay. That's what that's for. Okay.
Thanks, everyone. All right, Jason, we're turning it over to you to frame up the conversation.
Yeah, so
Nice boy.
back to what we were discussing. Sorry for that little delay. We're gonna cut genetic rescue down to about 10 minutes. So it's an important topic. You know, we've got a population that was once pan-mictic in a lake that covered most of northern Nevada. It's been isolated. Genetic rescue seems like a logical tool. So this is the time to start asking your questions for how we might move forward with genetic rescue and recovering LCT. I will start in the room.
Start in the room and then Jason will kick it to the folks on Zoom.
Yep.
Any questions, concerns, thoughts on potential solutions? Knowing that this is like an initial conversation, there will be many more.
Okay, this is Mary Peacock, and I just wanted to reiterate something that was said yesterday about when we go to do these reintroductions, this result that these guys have from Montana is really, really cool. But we also need to talk about, when do we do in basin, or within watershed types of transfers versus, what are we dealing with? Because this landscape is going to be, when you're talking about the Quinn versus you're talking about the Humboldt, the number of populations are going to be very different. Variation is going to be very different. And, there are examples from the literature where we're talking about a Florida panther and other situations where that was it. So this is something that I think we need to be thinking about for each one of these genetic or geographic management units. What are we really dealing with and how best to approach that?
So sorry, this is Helen Neville. So this is super cool, and it seems to me that you have evidence both of the matings that occur, higher fitness in growth rates and survival and things like that, but also mate choice in that it's the hybrid there are more hybrid families than single source families, right? So that is evidence again that if we put these fish on the landscape, they are actively choosing mates among the most diverse or different options, which is pretty interesting. So to me, it's like you try and mix and then the fish sort it out on the landscape in a lot of ways, right? Yeah, okay, good, it's nodding.
Jessica's got a question for you.
It's Jessica's names. Do you think there's ever a situation where introducing fish from like a small bottleneck population does more harm, like using them as a source for reintroduction does more harm than good? So it's like it's adding the genetic diversity, but from your results, it seems like they're not doing as well. So it's like, does the good outweigh the bad kind of in that situation? Am I making sense here?
Yeah, so I think what you're getting at there is outbreeding depression, which is essentially a decrease in fitness of hybrid individuals. And so, I mean, in this, we only evaluated the first generation of offspring, and we don't expect outbreeding depression to really take place until future generations. And so, yes, there is that danger of, hybridization between or among source populations being consequential. However, some of what Andrew's work has shown with genetic rescue, looking at multiple generations following hybridization among these populations that have been separated for a while, we aren't seeing that, right, Andrew? Yeah.
And the outbreeding depression is just caused because the population that you're taking from is just the genetic diversity is just so low. So it's still, no.
So it'd be like outbreeding depression. We could expect outbreeding depression to occur if these populations have been isolated for an extremely long period of time and have potentially developed local adaptations to specific areas. So like what Mary was getting at earlier with like translocating, populations among these basins, like, should we be concerned with potential local adaptations in the Quinn that aren't in, let's just say like the Reese or somewhere else in the Humboldt? And I don't know the answer to that. I mean, I think we should probably be hesitant with that, but there's a lot of questions revolving around that. Yeah.
Yeah, so I guess what I was thinking of is just in your graph, the population that had low genetic diversity, it was just that those fish didn't reproduce as well. And so like, is that still beneficial to introduce from a population that has low genetic diversity? If they're not going to do as well, but it's still increasing the overall genetic diversity.
Yeah, and I would say yes to that, as we do still see an increase in genetic variation in that population, and those fish still did reproduce, suggesting that there's some benefit of hopefully having that genetic variation still on the landscape, and we're able to, still capture that within reintroducing multiple source populations. Yeah.
This is Rachel Van Horn. So we tend to take, when we do introductions, at least in the Northeast, we take fish where we have the most fish to take from, not necessarily considering genetics at this point. So with one of yours, you couldn't take a lot because there wasn't a lot. So where is that line in the sand where taking more genetic diversity but less fish is more important than taking more fish with less genetic diversity?
Ding, 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 ding.
Right in the middle of the.
Oh, I thought it might be helpful to go back to FST plots and talk about that, because that's saying that each of those populations has unique variants. They may be bottlenecked, but they're bottlenecked unique sets of variations that don't exist elsewhere. And so if you don't include them in the buffer, you lose some total amount of possible variation that exists for species.
And one other follow-up on that comment is that I think in your study and in our situation down here, these populations are driven, the differentiation is largely driven by extirpation of other populations and drift. So this is not always necessarily natural selection that's driving these populations apart, but the remaining genetic diversity is simply somewhat random. And so we need to capture it, but it doesn't necessarily denote like these, extreme local adaptations, which is a consideration we need to be careful of, but.
And I just want to kind of bring this back real quick. That's the next kind of topic, is talking about drift versus selection, and the advantage of having someone who's not a geneticist moderate this session is hopefully I'll bring this stuff back down to earth, or at least for myself. But that's one of the things with LCT. There's 110 mountain ranges in Nevada. We've had tiny populations that are subject to drift or evolution or adaptation. It seems like if this were specialization or local adaptation, that we would have 20 species of cutthroat in Nevada, but we just don't. We see that they're very similar. And as Helen said, and I don't know if the people on Zoom could hear this, I don't know that we've answered the question if those FST or those difference values are related to drift, or if they're related to specialization. And so I think hopefully that'll generate some comment, and Mary's going to give a comment in the room here.
Okay, just a caveat, this is Mary. I do teach evolution at UNR, so I'm deep in the weeds here. But to Jason's point, talking about natural selection versus drift, when you're small, drift is always happening in populations. It's just whether it's a mover and shaker when you're talking about changes in allele frequency. But in small populations, drift is more the driving force in changes. The natural selection would have to be super, super high in order for selection to trump drift in those situations. So I think we are dealing with drift as the primary driver here, and possibly have been for a very long time, given, even in large watersheds, giving a metapopulation sort of dynamic of extinction and recolonization. So I think that is a harder thing to combat when you're dealing with drift, because there's no directed change in allele frequencies. It's just a random process, and you can lose adaptive variation just as much as you can lose neutral variation. There's no rhyme or reason to it.
And
Jason?
Yeah, go ahead, Laura.
Is there any chance you can click the chat box down at the bottom of your screen and pop it up? Because I guess folks are wondering if there's a way that the folks in the room can see the comments.
Yep, I got them posted up now.
The only one that has come up so far was Mandy saying there needs to be a commitment to monitoring, which is time consuming and expensive.
This is Travis Hawks. I just had a question. So assuming you have, you're working in a basin, you have multiple source stocks, and then you have kind of what Rachel was talking about, a endemic population that is small and isolated. Is there ever a time where it wouldn't be beneficial to do genetic rescue? Or like in the case of like a specific strain, like is there ever a time when it's beneficial to isolate that specific strain from maybe other strains of LCT?
Andrew, why don't you go ahead and take that one?
No pressure, Andrew, no pressure.
This is Andrew Whiteley. Can folks online hear me? Apparently that doesn't often happen when I talk. Oh, man, that's the question, right? That's when do we do rescue? When is doing nothing? How likely is that population to blink out if we do nothing? That's, I don't have the answer to that today.
So you have a larger population, but they're still maybe increasing fitness by adding. So you have a larger population and there's still the potential to increase fitness, give it a better chance of reproductive success, whatever. And you have another strain of LCT. Like how, I mean, is that just, would it make sense to do that? Like if you want to potentially increase the probability of a specific strain of fish to maybe just increase its fitness and boost its recovery chances. I mean, is that just how do you decide that?
What do you mean by particular strain?
I mean,
We're not dealing with.
my question is specific to say pilot peak strain, right? Where there's a lot of time and effort being put into isolating that strain. And is it possible that if you were to allow the contemporary strain to swim past Derby Dam with the pilot peak strain, you would have a better chance of reproductive success in those fish in the Truckee River? Or, I mean, are looking at the museum samples and what's available, is that not something we want to approach? That's just a big question I've had for quite a while.
Life history variation is a key consideration that I only glossed over yesterday, but in some of those West Slope sites, we found surprising amount of life history variation, even in headwater streams, where one of the two more northern sites, if you remember my graph, my map, where rescue didn't work as well. There were a couple factors at play there. One was that they had higher genetic variation. Another was that there was actually surprising life history difference, where the one I called Gold Run, if you remember, has a really long, slow life history for a tiny stream. It's colder. We're not sure why though. They're living till like 13.
Can you put the mic a little closer? You're starting to fade out when you talk.
And those didn't do well when we moved them to the other site in that reciprocal transplant. So there might have been some life history variation associated with that result as well. So I think that comes to play with LCT, where you have this history of moving to the big body of water and getting really big in a way that might not be at play with West Slope.
Before Mary responds, I just want to let folks know online, if you have a question or comment, or just make sure you raise your hand or start typing in the chat so we know, and I can direct Jason to pause to get your voices in the room. Right now, there's nothing, Jason. Go ahead, Mary, or wherever you were going next, sorry.
So with Rachel, what Montana's trying to do is not take more than 10% of a source abundance wise. So they've at least drawn a line where we don't, that's why only 10 fish were taken from that one source. Yeah. And I'm hesitant to go to here, but another layer we haven't talked about on the genetic side is the deleterious variation coming in. And so population genetics theory predicts that large populations harbor more deleterious variation. It's just masked in the heterozygous state. So all of us have enough variation that if those alleles were homozygous, we'd be dead like 3 or 4 or 5 times over. Small populations, either that deleterious variation goes away or it goes to fixation. And both happen. Can't rely, purging is when it goes away, but we can't rely on that. Fixation is common. So the worst case scenario is like with the Isle Royale wolves, where one individual brings in genetic baggage, does really, really well, and inbreeding reoccurs very quickly. So we want to avoid that in these scenarios, no matter how we set it up.
This is Mary Peacock again, and Travis, you know, I think what would be a really great thing to start tracking is in the lower Truckee, below Derby Dam, both contemporary and pilot are coming up to spawn. And they're using the same reds, and there's definitely interbreeding. You know, my data show that that's happening. So I think it would be useful to track that over time and see the impact of that kind of interbreeding between pilot and the contemporary strain. Because the contemporary strain, as we know, is from Summit Lake. And that's, you know, a whole different, I mean, it's a lake environment, but there's a lot of differences. So I think that would be a useful thing to do. And I'm adding to Andrew's work here. By saying, oh yeah, track that as well. But anyway, I think that's a great natural experiment happening for us that we can answer some of those questions.
That's only below Marble Bluff, though, not below Derby, correct?
It's below, at this point, below Marble, yeah.
Yeah, because they were segregating that Marble Bluff. is what you
Yes,
said
yeah.
yesterday.
But I think to address that, you know, do we start passing contemporary above the results from looking at below Marble Bluff could inform that decision.
So prior to the microphone being passed, the comment was that that's only happening below Marble Bluff. So yesterday you had a graph that was decades versus thousands of years. And it was like, it's better to do it over here in the decades column than over here in the thousands of years. And prior to modern agricultural diversions, like we aren't hydroelectric power here in Nevada, we are agricultural diversions. That's only 150 years ago that these fish couldn't make that decision for themselves. So is that the decades column, like potential, like, right, even, I mean, that's the whole Lahontan Basin, not just like the Humboldt Rit or whatever. 150 years ago, in a good water year, the Carson and Humboldt Sink connected still. So when we talk about moving fish, like technically 150 years ago, it was the whole Lahontan Basin. And we are now saying potentially only within watersheds. So that's just a question for the genetics, more of like in the mixing conversation, right? When you put that up yesterday, technically all of the Lahontan Basin is moving that way.
But not the whole Lahontan.
Well, yeah, not the whole Lahontan Basin, but
and so you've been.
Hold on, Mary, hold on. Take the microphone.
Okay, not the whole Lahontan Basin, but yes, good point. But there were, as the lake dried down, there were isolations that happened along that 10,000 year, you know, pathway. And those, we should be cognizant of those. But yeah, mixing between the Humboldt and the Carson via the sink habitat, you know, yeah, I mean, there's lots of things that we have artificially, obviously on the landscape imposed on these fish. But we have to think about that in the context of this larger hydrologic and geologic history, you know, when we start thinking about moving fish.
Well, and I would note one other graph that Andrew had yesterday that he was like, I'm not going to dwell on this, but I think it was a really important one, was the projected, and it's, you know, based on what we know about theory of isolated populations with given NEs and how quickly they will continue to lose diversity over time. So an important thing for people to understand is where we are today without any management action, these small isolates will inevitably continue to lose diversity. So we will be ratcheting down in the future without taking some sort of action to restore what would be ecological movement, not necessarily like changing of, you know, like you're saying, on a different time scale.
And hence the urgency for a genetics management framework or plan, because, you know, like paraphrasing Aldo Leopold, the first step in intelligent tinkering is saving all the pieces. And, you know, we were talking last night about how since, you know, from last meeting to this meeting, we've lost 2 or 3 populations. Like they continue to blink out in the absence of being able to recolonize. And we've altered the landscape so much that we don't get that natural recolonization like we used to. And so we need to save those pieces, but we need to do it in an intelligent way and figure out how to do that. So.
Okay. And one more thing to add to that, going back to Rachel's original question of, you know, like how many fish do we take from these populations and when do we, you know, say enough is enough? And I think in a lot of cases, we are dealing with populations. I mean, as we saw in some of those RIT presentations the other day, we have populations that are on the verge of being extirpated, you know, of 50 or less individuals left in the population. And I think when we're at that stage, I think we should be less concerned about preserving that specific population in that habitat and instead, you know, focus on conserving the genetic variation there, potentially moving that genetic variation somewhere else that it could be beneficial rather than losing it.
One other point that Andy made in the chat was that we need to commit to monitoring, which is another key. So I asked Andrew about this at AFS this summer, sort of what is the amount of information, knowing that we can't do pedigree analyses everywhere and follow reproductive success and all of this in such an intensive way, thinking carefully about the minimum amount of information we need to collect in the field to monitor and ensure, you know, the benefits are happening, but also the no harm side of it, that we're tracking any influences. And I know you have some ideas about that we could talk about later.
And I mean, so just to add to what Jason just said, like from a management perspective, like a framework would be awesome to where we can like look at the landscape and start assessing streams and saying like from our standpoint, okay, that one we can do genetic rescue and we can do this 5-year project, monitor, and then we can move to the next system. And like just going back to the whole idea of like what I think we're trying to do here and develop a way to go forward. And I think like that is crucial to most of the basins that we're working with.
Yeah, and I guess just speaking from my own here, not as the moderator, but one of the things that I'm concerned with as we go through this process, I want to make sure that we don't hem ourselves in so that, you know, down the road, because we know how it worked with the 95 recovery plan. We had a very prescriptive plan that put us in a box and kind of put us in a hole for, you know, 20, 30 years. And so I want to make sure that, what I'm hoping is that whatever this genetics management framework comes out as, it's not prescriptive. It doesn't box us in. It doesn't say only within this level of huck, or, you know, it's got to be kind of a living, breathing thing, or it's got to be at least adaptable so that we can do, as we learn more, the best for the fish across the range.
Absolutely.
And then I guess I'll just go into the next one. So we're talking about, you know, mixing fish and, you know, diversity. The other one is specialization. So we have some populations, like Dave Banks was saying in the Willow Whitehorse RIT update yesterday, that should probably be considered evolutionary significant units or DPSs. And so that's, I think, another discussion that we need to have is where do we go with that? Because we haven't committed to a, you know, an update of the recovery plan or anything, but we need to start having those discussions about, you know, what is a DPS? Where do we want to go? How do we want to manage these? And I'll just open it up if anybody has any questions about that. And then I'll get to Nick's question online.
Okay, you see that, Jason? Okay. And remember, folks online, if you have questions or comments, raise your hand or put it in the chat so we know to go to you.
So this is Mary again. From the work that we've done looking at when we wrote that book chapter, it was clear that Willow Whitehorse stands out. And so we absolutely need to be thinking about separating, well, I don't want to say separating, but we need to think about that as a unique unit. It's not part of the Quinn. It's very different, and the hydrologic history of the basin supports that. So yeah, I mean, yeah, morphologically, genetically, everything. So, yeah, it's definitely different.
And so what are the risks if we go into the DPS designation? I mean, certainly like for Oregon, there's a benefit. Like, we could potentially delist LCT in most of the habitat in Oregon. But what does that do?
I mean.
Well, no, I'm just saying, like, hypothetically speaking.
The white horse shows that, and I think we've all learned over these recent droughts, that the habitat there is not as resilient as some of it, as we've been assuming in the past, right? So look at what Robin presented yesterday relative to the Hawaiian Islands, like, yes, the demography is increasing, and yes, there are signs that these populations are improving, but there's too much risk given the isolated and, irreplaceable nature of some of those habitats. And I think one thing to consider is that Willow White Horse is a big interconnected system, but it's one watershed. And it's drying and it's increasingly at risk. The genetics demonstrate big bottlenecks in there relative to the number of fish we see. So, I mean, those are still factors that we would need to consider, I think, relative to the range-wide loss of something like that. And it would open up a whole other process for all of these other populations too, but all to consider.
Yeah, and then I'll get to Nick's question really quick here for the geneticists. Are there any demographic indicators associated with inbreeding depression?
Part of the slide I showed yesterday, so I showed that in one study, we've looked at inbreeding depression in a small isolated West Slope population where reproductive success declines with inbreeding. We did pick apart different demographic, and I didn't show it yesterday, but I'm looking at a graph now that I'll explain real quick. We see a negative effect of inbreeding on family size, survival to age 1, transition from subadult to small adult, transition from small adult to large adult, and then the probability of reproducing. So we do see it across vital rates, but whether you could just pick one of those to monitor and get a full picture of inbreeding in its population is unlikely.
And you would have to track those families through time, know their origin to be able to compare those to see if a quote unquote hybrid or a translocated or or if not inbred or inbred would be different within that population.
Yeah, to kind of get at Helen's monitoring question and what it's an open, do we need to do rescue in a way where we intensively monitor still in a new subspecies like LCT? I would argue probably, but I think where we're hoping we can get to is more rescue across the landscape monitored less intensively. And the best way to do that might be to look at migrant ancestry over time and kind of pop into a population periodically. Just genetic variation alone, I don't think does it, because you could get a boost in genetic variation and still get outbreeding depression, and paradoxically. So if you could look at genetic variation and migrant ancestry over time, I think that could be done in a set of less intensively monitored populations.
Jason, I have my hand up.
Oh, go ahead.
My role here quick, but I just wanted to say, thinking about the DPS question, when we did the situation assessment in South Fork Humboldt, from a social standpoint, people were very interested in the notion of having these distinct population segments because, and then they would be more willing to individually and collectively take on some of the risks and costs and potential consequences of reintroducing fish or having better habitat or whatnot, if they knew it was going to impact their community, their area of interest. Being tied to all these other areas and what happens or doesn't happen in those places created a little bit of a disincentive for folks. So I just wanted to throw that out there.
This is Chad Ellison. Another factor we need to think about if we went down a DPS route is the designation of critical habitat, because we would be asked to do that if we split it up again. So that is another thing to consider. I'm sure folks would not be too pleased if we went down that route. So there's pluses and minuses of doing it. But just something to think about.
Yeah, this is Matt Campbell. When I see like life history variation and you have this interplay between like migratory and resident types. I was wondering what the status is for understanding the genetic underpinnings of that variation across original Hottons and if it's like conserved. I just don't know. I haven't seen much in the published literature, so this is like a question for like what's out there that people know that I don't know.
Yeah, I think the short answer is we don't know. Yeah. There have been some alleles, right, that have been kind of annotated and linked to certain processes, but yeah, okay. Yeah, a comment in the room was just that we don't understand LCT genetics quite as much as other salmonids and salmon.
Yeah, because I think this goes back to what Travis brought up earlier with like outbreeding depression and life history ties, because it may be that a lot of stuff is strictly theoretical, but if there's like a single like underlying block of genetic variation that contributes to this like life history variant, but it's like super favored, then through introductions like a few individuals that can be quickly selected for, or if there happens to be like hybrids that are formed between two different strains within that, it may, that like it still be favored by selection and it doesn't really matter too much. But in the case where it's a super complex trait that's broken down by that hybridization, then there's no benefit to it.
I would say, I think that's definitely worth looking at. Again, in an earlier study, we tried to do this looking at outlier methods across the genome, and the stream populations in particular were so bottlenecked that we just couldn't get much of a signal. Things have advanced a lot even in the last 8 years, so doesn't mean it couldn't be reevaluated.
This is Andrew. I'm hesitant to keep saying in West Slope, this is what we see. But we did a genome scan recently that we're going to publish soon, where we looked at isolated and non-isolated populations, and we looked at 300,000 locations across the genome, and nothing major emerged as a genetic. So not unlike in the salmon world where these major genes of major effect are emerging, we didn't see that in isolated cutthroat. Probably not the same underlying genetic basis to life history variation, maybe in West Slope, but as I just said, we did see surprising in a couple populations. So,
and one other facet of that is, I think you mentioned yesterday, Andrew, that you're seeing age 0 fish reproducing in some systems where they have the possibility. Jason Dunham's done some work on fire and rainbow red band trout populations where they see these immediate shifts in life history to take advantage of new habitats. So they'll start breeding at age 0 if possible, or, change that kind of reproductive life history. And I would suspect, based on what you just said about Westlook, that these fish are really plastic in those instances for dealing with this very dynamic habitat. So it may not be like the conserved regions and anadromous fishes, for instance, but they preserve a lot of response landscape, I guess, but who knows?
We got a question on the Zoom real quick, Chris, and then we'll get to you. Go ahead, Nick.
I mean, it just kind of sounds a comment leading into a question. It sounds like we're talking about putting Humpty Dumpty back together again. And the real question with the genetic mixing is what Humpty Dumpty looked like in advance before it got broken up. And the question here is, how much did these fluvial populations move around? I don't work in the Marys River at all, but it sounds like there's pockets of intergressed fish that just are not moving too much. They're staying hunkered down and put. Was there a ton of pre-existing genetic structure on the landscape or population structure that, just within basin, looking at Mary's talk yesterday with the Truckee River system, you had 2, maybe 3 genotypes that were discernible and present in Tahoe, Pyramid, Truckee, Upper Truckee. To what extent did that exist on the landscape before? Is that a question we can even ask? Sorry, it's kind of a long one, but we need to know what our target is. What are we trying to reconstruct here?
Yeah, I'll let Mary answer that.
Okay, this is Mary. That graph showed genotype clusters of different colors. It wasn't just one genotype that was defined by those clusters. And I think that the lesson from the museum samples in the Truckee and from Helen's work for her dissertation shows that there was in these watersheds a lot of movement. And we tested a metapopulation dynamic. It doesn't absolutely fit that, but it definitely fits this a mix of metapopulation and life history variation. So there were fish that were resident in these headwaters and fish that moved around in the main stem rivers and yes, and we also radio tag fish and followed them around. And there was, you know, we did see fish moving in that main stem Marys River. So I think, you know, yes, it's a sample size of 2 from our museum specimens and from the Marys River. But I do think that this is how the fish survived in this highly variable high desert environment, is these large interconnected systems where they could risk spread across this landscape. And we do have evidence that in one of the streams in the Marys River during a high watershed or a high spring runoff event, we lost the upper resident population, but that it was then recolonized years later. And we saw this through the time span that we were out there sampling fish. So we know we have some limited evidence that that absolutely happens. And so I think that should be our goal, is to try to recreate that dynamic for this fish that will then, yes, and Helen's work on Maggie Creek is doing just that, reconnecting the streams, and we've genetically tracked how they move once we remove those barriers. So I think that our goal needs to be to try to recreate that dynamic and then let the fish sort themselves out on the landscape. We just need to give them the tools to do that. And I know that that's a big, tall order, and we can't do that everywhere, but this talk of genetic rescue is to try to like do that in baby steps where we're not losing unique genetic variation within a basin. But ultimately, in all the cases of genetic rescue that you look at in the literature, the take-home message is that the habitat has to get restored, or else you're just going to be back in the same position down the road. You know, genetic rescue isn't a panacea, as Andrew said many times yesterday. We need to deal with habitat issues.
This is Dave Banks with ODFW, and I'm just thinking about this conversation about this. So an example we have in Oregon with red band trout, so still not Lahana cutthroat trout, but maybe goes to some of the stuff we'll talk about when we get to the habitat stuff later today. But we have a river where there was a reservoir that was created at the bottom that recreated some of those lake lacustrine habitats. And so prior to that, we didn't see an adfluvial strain or type of red band trout, but you built the habitat and it reemerged. And so maybe that goes to, well, the habitat, you know, where we can, you know, we have problems, we have isolated populations, it's not always possible, but where you can reconnect that habitat, then you'll get the variation of the different types of life histories.
Jason, do you see the comments in the chat here?
Yeah, I do. Go into Travis in the room and then we'll get to that. And
Okay.
maybe Travis, while you're on there, if you want to address these.
They're talking about you specifically, Travis.
You don't have to go ahead with your.
Okay, so sorry, this is just back to the whole DPS thing, and it might be a question for Chad left, but I would assume that if, you know, we went the route of DPSs and had the possibility of delisting DPSs, that also brings up the possibility of uplisting DPSs, which I think there would not be a huge appetite for that in some of the stakeholder groups as well. So it's just another thing to keep in mind.
Good point for discussion. And I'm gonna go to Roger's comment here online. He just talks about how there is some natural reproduction below Marble Bluff, which we mentioned, but we don't know the recruitment. There's a lot of fines down there, but they're gonna look at admixture. He mentions that the pilot strain is unique and it has a huge difference in life history strategy. Older at spawning age, not so much the case for the contemporary strain. High levels of heterozygosity, mixing may not be beneficial, may actually be damaging. Mary, do you want to comment on any of that?
Yeah, the Pilot Peak is definitely a unique life history, and the contemporary strain doesn't have some of that. They're different. However, the Pilot Peak contemporary strain was founded from a very small population, and we have worked really hard in our hatchery management or the genetics management to maintain the heterozygosity that or maintain allelic variation that is in that broodstock, but it still was a small population. So and even though we're working really hard against it, you know, if you put those fish on the landscape, you're still dealing with a smaller amount of genetic variation than in some of the populations, not necessarily in the Western Basin, but we only have a fraction of the historical variation that was present in the fish that were in Tahoe and Truckee and Pyramid. So I think going back to the thing, they are mixing with contemporary in the lower river. So it'll be interesting to track that and see what impacts that has.
So this goes back, this is John Paul with Fish and Wildlife Service. This goes back to Mary, your talk yesterday. It appeared that there's a plausible explanation that there were multiple life history strategies in the Truckee system, or in many of these systems perhaps, but particularly the lacustrine that was in lacustrine that was in both the Tahoe and Pyramid Lake. And so likely there were different strains based on different life history strategies and
Different life history strategies for sure.
right. And it sounds like, you know, we need to learn a little bit more about maybe contemporary has a different strategy than the lacustrine, and so perhaps there's room for both of these to coexist. And so that's part of the conversation, but we just don't know if they do hybridize or.
Well, they definitely in the below Marble Bluff, the genetic data below Marble Bluff, there's only limited spawning habitat. And so contemporary and pyramid are coming up into that lower habitat and using that to spawn. Right, yeah. And so there is a hybridization between the pilot and the contemporary. We just haven't tracked those individuals to see what the consequences of that are. And I would suggest that we start to do that. And I think Andrew's group is going to start doing that, because we don't know what the outcome is there. You know, it's a total unknown. We haven't and so I think the questions about what's the role of contemporary in the future versus the pilot strain in pyramid or in the Truckee River system is an unanswered question. And we're not going to be getting rid of contemporary in the lake. And so if our aim is to have a naturally reproducing population there, this is a question we have to address, because, you know, they're in there and those fish are going to mix. And, you know, there might be some mate choice that goes on down the road, but those are questions we're just not nearly at the level that Andrew's work with Westlope. But we should definitely be going in that direction to answer some of these questions, because this is a big issue. You know, the Pilot Peak are a unique strain. They are this historical strain. But like I said, you know, they were in a small stream for 100 years. We captured a fraction of the variation, genetic variation, at least at neutral markers, that was likely when we compare to museum samples, that was likely in this large population. So what did we capture? I don't know. You know, is this ready? So they grow huge. They have these features that were prized before they went extinct, so or were extirpated. So, you know, these are really important morphological and life history characteristics that we want to preserve for these big lacustrine populations, but there's a lot that we don't know.
And going back to policy too, I think to add on to those comments, I don't think the question of and Mervin's not here, so I don't want to unfairly characterize this question yesterday, but I don't think that the question has been resolved as to how the contemporary strain contribute to recovery. And so that's kind of a question that needs to be answered first. And we know that in Pyramid Lake, those contemporary strain add to the recreational capacity for that lake, which provides income and incentive for, you know, the tribe to have fish in that lake. And it's a really complicated social discussion that I don't think we can just write off and say, well, this is the original strain and this is what we need. So
I think a really important sidebar to add as we have that conversation about what sort of conservation benefit could contemporary strain contribute to recovery is also knowing the genetic makeup of what a contemporary strain fish is. Is it one that comes into Marble Bluff that has adipose fin in early March, the same fish as a what we call contemporary strain that is an adipose present fish that comes into Sutcliffe spawning channel in mid-April? You know, I think it's important to also keep in mind rainbow trout admixture into some of the fish that are in Pyramid Lake, and really know what we're talking about when we have this conversation.
And Lisa has a comment in here about wanting to preserve the genetic legacy of the pilot pig strain and understand their use of the habitat, reproduction, and recruitment.
anybody else have anything on the genetics? Otherwise, we're going to move into the conservation efforts database.
Lisa added the Fish and Wildlife Service supports the tribe's recreational and historical program and for the lake population of their contemporary program.
Okay.
All right.
Move on to the conservation efforts database.
Thanks, Jason.
Yep. Are we going to take a break?
No, do the
CD. Okay, yeah.
There it is.
All right. Hi, everybody. My name is Leif Wickman. I work for the USGS in the Land Management Research Program, and my role here is I'm part of the CED team, along with Matt Heller from the Fish and Wildlife Service, who's going to be talking. We're going to do a little bit of a double Dutch here, do-si-do presentation style. But it's nice to actually see you all in 3D. We started this project 2+ years ago during the pandemic, and so we've seen a lot of you virtually. So nice to see everybody in person. But we're just going to provide a quick update on kind of where we've been, where we're at, and where we're going. And I'm going to move expeditiously. So these are the people who are on the CED team. Here's our timeline. So we've kind of parsed this out into a phase 1, phase 2, phase 3 sort of thing. So back in the winter of 2020, we started setting everything up, getting agreements into place, establishing sub teams, working with you all to replicate what was called that requirements document. What are the needs, functions, and purposes you all wanted to get out of this tool? And a lot of what we're talking about today is data. I mean, there's a lot of data being collected. And so what we're trying to do here is bring that data together and make it work for y'all. So one of the big things we did in phase 1 is we established 4 sub teams, and that's kind of fitting in line with how this meeting is going. We've got a sub team that talks about demographic data. We don't have a genetic data sub team really established yet because we've been waiting for a lot of the conversations that y'all are having today, and that's something we'll move forward on. We've got one for conservation actions, and then of course the Habitat Core Team, which Robert leads, and we'll talk more about that this afternoon. In phase 2, so fast forwarding to 2022, we continue to work on development. We'll talk about some of the highlights out of that. And really this is intended to build you all a prototype, something to swing at. And I'm going to turn it over to Matt. One of those big things we accomplished in phase 2 is kind of our spatial framework. And so I'll let Matt talk about that.
All right, thank you, Leaf. Yeah, so we have the banner there covering up a little bit of my text there, but anyway, so the spatial framework, I'll start with that. So the CED collects data, and whether that's spatial data, tabular data, or that kind of that middle that field report data, that's kind of a combination. And we came up with a nested hierarchy. Some of this I've talked to some of you about in the past, are presented, and now we got some data to put with it. So right here, we have the first kind of that base level, or that fine scale level. That's for a lot of the field data that will get rolled up into the other the higher levels of the spatial framework. So we have a start on sample points. You can, I'm not sure if you can see on the map there, we have some gray crosshairs. And then we also have streams and lakes. And this is showing the occupied streams and lakes that from the good work that Faith handed over. And then we also put in the not occupied also. So this is that fine scale. We're not going to necessarily have much summarization at this scale, maybe some very basic summarizations in the future. Then there's the spatial reporting unit framework layers. So this is going to be the base of all the reporting and the summarizations, and all the other GIS data will be tied to these spatial unit framework layers. So the population reporting units, that's the one that I guess it'd be the main one that we're focused on here, is shown in blue. So many of you were part of on a team with Faith to create these what we're abbreviating PRUs. And then we're showing here now in tan, the interconnected population reporting units. And then we have broad scale framework layers too. So this is exactly what it sounds like. It's more for general reporting. And you all probably are very familiar with these areas here. These are the management units and also some of the subunits. So we also have an area for the range and the project area. So we just talked about the spatial reporting units. So you saw we made progress on that, creating the data. And we have that draft complete. Then we have these other layers too. So they're based on the sub-team input and their needs. So with the conservation efforts, we have that ready for all users to test right away now. We have the web editing capabilities for y'all to test. Many of you heard it, and Leaf's going to bang that drum some more here. So here we have a couple just test polygon, not even, I guess, very crude test polygons here. They're kind of those Easter egg colors, but we can also do points and lines for entering in the conservation efforts. Next is the demographics. So we just have a start on that. We have the sample site. So that's just the base and much of the other related data. So we'll be getting, this will be getting more evolved in the CED as time goes on here. So over the coming months. And then the habitat, we're in touch with Cash and Wally with the sub team for the data handover to get the remote sensing and derived products. So this habitat component of the CED will probably be a continual one that would be going, for 6 months, a year to come, and we'll have that available and rolled in. And then we'll be staying tuned with the genetics folks too, when that and then we're estimating that to be phase 3. So you can see, we have this. I'll talk about it a little more here in a minute, but we have a beta site that will be rolling out for a public website here in a couple weeks. All right, back to Leaf.
All right, so this is kind of a snapshot of where we're at now. So back in January, we rolled out that beta version, and we asked you all to start beta testing it. I can't understate how critical the beta testing is. We have what are called one-to-many relationships. We have dozens and dozens and dozens of lookup tables. And as Matt described, we've got this spatial hierarchy. So when data is entered at a stream or a segment or a survey point, we can roll that up and summarize it and use that to evaluate effectiveness of conservation actions at different scales. Because of all those different relationships, there are bugs aplenty. There are breaks. We need your help in finding those breaks. There's probably 25,000 different combinations of different entries you could do. We can't test all 25,000, but we can test the ones that you think are going to be most common. And it's all fake data. So all you have to go in is just type in test and you make up information. After you walk through it, you'll realize you could probably create a test record in under 5 minutes. Especially if it's just made up data. And we will, I should have said, we're going to send an email to this group. It'll have a PowerPoint of these slides. It's also going to have the beta testing links if you're within the Department of Interior, if you're outside of the Department of Interior, and we've got feedback forms. We've got a guide showing you what are the things we'd like you to test. So we'll share that with you all. And that can even go down to, I don't like the way this is phrased, or you misspelled this word. I mean, there is nothing too small that you can't point out because we want to make this right. So we're in the middle of the beta testing phase. And so if you haven't registered, please go to the CED and register for a user profile. And again, this first beta testing effort is focused on conservation action entry. We also have the ability to view the summary of demographic information. And then Justin created this project narrative, and it's almost like a field journal, things that you can enter that don't necessarily fit nicely into the CED, but a way to record that information. We can also view and summarize some of that raw demographic data, but we don't have the ability to enter it in right now. So again, this is kind of an overlook at the beta testing rounds. So round 1 is done. And I'll be honest, I think we had maybe 2 or 3 test records in that first month. So we're really going to impress upon the group the importance of that. So in a couple weeks, we are making some updates to the site. We'll start round 2 of beta testing, which will go from about mid-March until the end of April. Then we're going to take May to make adjustments and respond to the feedback that you all provided us. Beta testing round 3 will be in the month of June. That's going to focus on, we'll be adding additional functionality as we go along through the beta testing. So if you're thinking, I'll just wait until June, well, that just means we're going to have 15 things to test instead of 5. So if we can do this in an iterative fashion, that would be great. And again, those will be the links for the feedback forms. So again, this just kind of, I'll go over this pretty quickly, but it's just anything. General site navigation. I went to this page, it didn't work. Again, just literally anything you find that you want to provide comment on, we want to hear about it. And I'll turn it over to Matt.
Yeah, so this is kind of what's been happening, the functionality in the phase 2. Again, my text is covered up there. So full mapping capabilities across all components. The first draft is complete, and big thanks to Faith and all the crew that we're working with her. Great to team up on that. And we now have ArcGIS Online services available. In the next coming weeks, we'll have the file-based downloads for the data that's available to download that's not password protected. And the public interactive map, like I mentioned, in a couple of weeks, we'll have the public interactive map that will be available for testing. And that will have search and query. It'll be the one-stop shop for the conservation efforts and some demographic data, and then some basic summary reports. So and that's just the public map. The password protected, all the data entry pieces, that's been live for weeks now, like Leaf mentioned here. So there's other things that can be tested right now. And then also, yeah, like I mentioned, we're also in touch with the Habitat core team to get their data uploaded and be able to view that in the viewer or in the public map. That's probably more a month or two down the road. And then we'll be staying tuned with the genetic folks too. Back to the same timeline here. So I think Leaf, you probably talked kind of the winter. Now we're getting into the spring. So we're going to be continuing to beta test. We'll have a soft release of everything, some training materials, and we'll be doing starting up the public outreach come the summer for the official release. And we'll be conducting trainings, and we'll be looking at other authorized decision support tools that we can integrate with too. And then in phase 3, we'll start in the fall. And that's when the hard, well, I don't know if hard work, but a significant amount of work for all you data providers. That's when we're going to want to be collecting data, and then also working with other tools to integrate with that. So this is this slide is talking about phase 3, what to expect for functionality. So we're going to integrate so in this early phase, we have the demographic and the conservation efforts. We're going to also integrate the genetic, what we can get, and habitat information. So, and what does that mean? So that'll be for all you data providers, you'll be able to link that information or those kind of themes up more. And then for the public user, they'll just be able to make sense of it all on the public interactive map and some other web resources too. So integration of all those different components. And then we'll add more summary graphics, data visualization tools, more summary reports, more data and query export capabilities. And that gets part of more maybe down the line in the user testing, where you can suggest, hey, it'd be nice to have a particular summary report or query. And then finally, refined data provider editing tools too. So that would be another thing that we welcome your input on if something's just is a little clunky when you're trying to provide data. And that is it. It's open for questions. Chat.
Great progress, thank you. Wait, so can you clarify, sorry if I missed, who did you release the first round of input? Was it just the Habitat group, or we supposed to be giving input that I don't think I ever saw?
It's open to anybody with access to the internet. So we've been announcing these on our core database team. So end of January, we held that call. And so I'm not sure who is all on that team.
I can speak to that. Yeah. So similar to the Habitat core team, we have a database core team, and then we expanded that group out with a smaller subset. So it's greater than the database core team, but smaller than the 200-person interagency list there. So we had representation from, all the parties there on it. It just wasn't that 200-person wide blanket email on it, just to get that first round of the beta testing on it. And now as we roll into phases 2 and 3, keep expanding that group out to bigger and bigger for more feedback on it. So, yep.
Yeah, Neil, and that's a real good point. We'll work with Sean and Faith and maybe get that out.
Sorry, I just want to make sure.
No.
Dave Banks. So Helen, you weren't the only one. I had no idea there was a phase 1 going on, or that beta test. So I know you talked about early on, you kind of had all the agreements in place. We've had an update with some stuff in our state that legally requires us to do stuff about data sharing. So how do we go about updating that? And we have recently, so we have an agreement with the U.S. Fish and Wildlife Service, so I don't think it's an issue, but how do we update if things change for various partners?
So if the state needs some sort of data sharing agreement with USGS, we can certainly set that up. The way that as you notice, we've got a .org for the website here. So populating the CED is voluntary. Giving information into it is voluntary. And so that has been a way that we've kind of avoided having to have state agency or agency specific data sharing agreements. But again, and we work with Oregon on sagebrush and sage-grouse stuff. And so that sometimes there's concerns about data sensitivity. I probably should also state that everything entered in the CED is password protected. And so that demographic sub-team, the habitat core team, the genetics sub-team, and the conservation action sub-team, they get to determine what the public gets to see and not see. So like your empirical monitoring data for demographics, you'll get to decide what level of information is summarized. And so that's how we make sure that everybody's comfortable with the level of data we're sharing.
Yeah, one other comment too. Yeah, and that's why when the slide that was showing the timeline on decision support tools, so that's why I use the term authorized decision support tools. We're not just gonna let any decision support tool have at it, so.
I just wanted to give you all kudos because this has really evolved into something very impressive. I think it's an incredible tool for really building the collaboration around this effort. You guys are really knocking it out of the ballpark. I also want to make sure everyone knew that today is Leaf's birthday.
Happy birthday to you. Happy birthday. Happy birthday. I don't get uncomfortable speaking in front of people, but y'all, you know, I feel pretty uncomfortable.
Your face is red.
Yeah, sure it is. And I also want to acknowledge, and I think he's online, but Justin Welty is a member of our team here too. And he's the lead plow horse on this. Matt and Justin are the ones pulling the sled. And I also want to thank all of you. We've got our sagebrush CED and it's been up and running for 9 years. And in 2 years, this group has really pushed the envelope, adding the genetic information, the demographic information. And once this thing comes online, it's really going to make folks in the larger sagebrush ecosystem really look like, are we doing as much as we could to tie all this information together and help evaluate effectiveness? And that's truly what this tool is, helping to evaluate effectiveness. So we're putting the limited resources in the right places. So just a big thanks to all of you. And I also should mention, thank Helen and others. Trout Unlimited provided funding to help get this thing off the ground, and then Fish and Wildlife Service Nevada Field Office has been providing to help us keep going. So just a big thank you. This group has been great to work with.
Leaf, next time you talk, can you adjust the mic up here because you're kind of quiet?
Yes, ma'am. I'm probably just still embarrassed over the birthday.
I know. I could tell you were shrinking violet right there.
All right.
Anything else from folks online? I just want to make sure you guys saw Lisa's comment going back to the pilot strain conversation about prior to reintroduction of the pilots in pyramid in 2006. Their documented first attempt to reproduce 7 years later, there was no documentation or evidence of contemporary strain spawning. So hybridization with pilots will be limited impact.
Can you address that, Lee?
I know it's a going back.
All right.
Well done, thank you.
Thanks, Leaf and Matt. We're going to take a 15-minute break here and come back with the Habitat core team on it. And just to follow up, make sure everybody's aware, there were 3 presentations today that were originally scheduled to be given, that we're going to break those out and do as a separate virtual workshop sometime between now and mid-April, probably in that early April timeframe there. So everybody on the interagency list will be seeing an email invite for that virtual workshop coming up on it. And to follow up from the genetics discussion, that was just the very early on to, you know, start collecting some feedback and get thoughts where everybody's at and stuff. Will not be the last that folks hear about those conversations and stuff on it. And Faith and Catlin were working on capturing all those comments and thoughts that folks were sharing, and all that will be shared with the meeting notes from the meeting here. So see everybody back at 10:15 for the core team, Habitat core team.
Cody, before you disappear, can I chat with you for a second?
Yep.
I heard Leaf or somebody was talking when they were giving their presentation that their text was covered. Is that the picture? I just want you to make sure you know that if you go up to the top right in that view,
Okay,
you can change it to just like a speaker view or a gallery view. You could minimize the number of boxes that are open if that happens to be part of your problem, or, you know,
yeah.
check, don't show non-video participants or whatever. And then you can also move the picture boxes around if it's blocking people's text. So I just wanted to make
Yeah,
sure.
we got that one. It's periodically the one, that has the taskbar with share and all that stuff will pop up.
Oh, okay, okay.
For some reason, sometimes I think maybe the cursor gets bumped and it activates that because it kind of comes and goes.
Okay, yeah, there's nothing you can do about that. I just wanted to make sure you.
Yeah, we got the other one kind of up over to the top left corner where seems to be the least common.
Yeah. Perfect. Okay, thanks.
Cool.
All right, we're gonna start again in about a minute. Cody, are you ready? There we go.
Robert's up first, and that's his right there. on that.
Okay, are they going one after another?
Yeah.
Let's pull them. I already informed him of that yesterday.
I'm gonna try to keep us on time. I'll just give you like a 5 and a 1. Should be pretty good.
Oh, yeah, I corrected on the one I sent it to somebody else.
Alrighty, we're gonna go over to the Habitat core team on it, and we got about an hour presentation with all the members of the core team here, and then we'll be doing our lunch break here. So here's Robert.
I work for the USGS. If you've been to these meetings in the past, we've been talking about this for a few years. Acknowledge my co-authors at the USGS, Utah State Fish and Wildlife Service, in Ken at the UNR. And also want to acknowledge a number of people who help out with this effort. The Habitat core team, we meet. Sometimes it seems like a lot, but people have been really good at contributing. Ranch managers and owners gave us access to lots of private land this year, funding from BLM. And now Cody, Jacob, and Mike have been really awesome for us in terms of like logistics, and it's been super helpful. And then folks at the BLM. Everybody's seen these kind of slides, right? Massive reductions in distribution from historically. And LCT have been listed for a long time. Habitat's identified as one of the factors that led to their decline, like many other salmonids. And yet there's been no consistent status assessment, right? So how can we prioritize how to move forward with one of the few things that we can do in the world is try and enhance some of the habitat. And so this is going to be increasingly important if you think of trends in climate. So this is just temperatures across Nevada from May to August for the last 40 or so years. You know, clearly it's not going down, suggesting so it is getting warmer. We all know that. And this is from the Nevada Climate Assessment, which also predicts it's supposed to be getting wetter. But despite that wetness, increase in wetness, I get this transition, it drives me fucking crazy. Despite the increasing wetness, this increasing temperature is still going to mean less water, right? So people see the Nevada Climate Assessment, oh, wow, it's going to get wetter. It's going to get warmer, which is going to offset most or all of that increase in wetness. This assessment is not a referendum or assessment of land management activities or their impacts on habitat. That is a whole nother issue, and we're not doing that. And so this is an assessment of the stream and riparian status as it relates to attributes important for Lahontan cutthroat trout, but also riverscape health. What do I mean by that? And there's going to be Cash and Wally are going to talk a lot about this. But really, if you look and you think about a lot of the habitat studies that we do, there's just so much focus on, you know, what percent pool is there and just in channel attributes. Yet we know from literature, past studies, that the floodplain is a big driver of in-stream habitat. And it's not just the location where we sample or adjacent to it, it's the floodplain and the riparian conditions upstream of that location that are also contributing, fine sediment, large woody debris, et cetera. So we're so focused on these, like what's going on right here, but we know we need to do so much more. Land management, climate effects, you name it, are happening everywhere upstream of these locations as well. And so we're trying to take a different approach in terms of assessing LCT habitat. The extent of our assessment is to, first, we're using, Faith was supposed to give a presentation beforehand, the climate suitability information that was a product of Trout Unlimited and Jason Dunham's work, where it kind of looked at the historic distribution shown in gray here and said, hey, there's a lot of areas that are highly unlikely to be suitable for the species in the future. So those are not part of this assessment, right? In black is what was deemed, again, by TU's work and Jason's and Faith's, is areas that are potentially suitable, right? There aren't LCT there, but these are areas that may be suitable under certain potential habitat improvements, reducing limiting factors, et cetera. But the extent of our focus is really on the currently occupied, with the stress of trying to answer questions at the management unit scale or subunit scale. So I have the subunits from the Upper Humboldt separated here as well. So we're trying to address them at the subunit scale, but we will have information also at the population by population scale to give some information. Again, we're trying to do something different. If you think of AIM, other programs, they do a great job of looking at reach-based stuff. But again, we don't have enough money or time to focus on reach-based assessments of field surveys. So we're trying to integrate different sources of information, from field surveys, the stick and tape, to using drones, GIS, and remote sensing approaches. One of the first things we really want to do with this is to provide some context of what the landscape is like. So for example, here we're using GIS approaches that Utah State and Wally and his crew have developed to get at things like valley bottom confinement. So how confined are these streams? So yellow is the suitable, this is the Upper Humboldt, obviously. Yellow is the suitable. Colors from green to warmer colors go from more unconfined to confined. And you can see a couple things. There's some obvious dramatic differences from the lower Marys up here to the headwaters, where the lower Marys is really unconfined system. That contrast is helpful, but it's also interesting to look at how much variation there is within populations, like, I can't see it, like in Hanks Creek, right? There's areas that are unconfined and confined on a tributary, right? We also see variation in tributaries themselves, across tributaries. Hanks Creek's right there. Across the way is Current Creek, unconfined valley bottom, still a tributary. We're going to have very different expectations for habitat and approaches moving forward. We're also combining that with information on channel slope. So how steep the landscape is. Again, channel slope, important driver of what we would expect given its impact on things like stream power. So merging these two things together gives us a template for a variety of things. First, it reduces the amount of time we have to spend in the field measuring these things. So programs like AIM and others are measuring channel slope, like using using stadia rods and transits, super time consuming. Why are we doing this in the field when we can get a pretty good estimate using GIS approaches? This also really affects our expectations of stream habitat. For example, if we imagine three different landscapes, from confined or laterally confined on the left to partly confined and laterally unconfined on the right, and you look at the pictures down below, you see, we would have very different expectations of what should be there if we're assessing habitat in confined systems where the stream has very little access to the riparian area, right? So our expectations are really different there in how we're assessing status if we contrast to these kind of more unconfined areas. This also affects our thoughts on, like, what is our potential or capacity to increase the health of these areas. And in confined areas, right, there's not that much we can do laterally. We can grade systems back up from restoration actions, passive or active. But if you think more places like that are unconfined, you have the capacity not only to bring the river up, or grade it back up, but also laterally providing more water storage, right? More riparian production, et cetera. So having this context, we think is quite important. We started field sampling this past year in the Upper Humboldt in green and the Little Humboldt in purple. Upper Humboldt alone is a massive part of the current distribution of LCT. These two areas account for about 39% of the occupied habitat. So we had a pretty good chunk this year, or this past year. And I'm going to focus just on the field surveys and just some tidbits of information that we have from that. We will have our annual report done. It's just about done. We have a few more things to do with that, and then we'll send it around. Cash is going to follow up some discussion of drones, and then Ken is going to talk about GIS approach, remote sensing approaches of NDVI, and Wally's going to finish up with kind of tying in some other measures that we've been working on. We have, for the purpose of our annual report, we've also assembled, we met with NDOW last year in Reno, and we talked to them about utility of past JAWS data. So we've been working to try and assemble past JAWS data. There is information out there. Let's not throw it away. And so just for example, our sites that we sampled are shown in green, the LCT HSA habitat status assessment. In black or dark gray are areas that JAWS has data from 2010 moving forward. In orange are the AIM sites. I don't want to spend a ton of time on this. A couple things that are worth noting. If we focus just on one approach, JAWS, we're not going to have a lot of information across the landscape, right? So we don't have a consistent way of looking at habitat. You can also see this with AIM. Not a lot of sites on the landscape. AIM is meant to ask questions at really big scales, not at scales relevant for populations or addressing limiting factors for populations. Assembling all three, though, you can see we do have a fair amount of information. We didn't finish some parts of Lower Marys. That's why there are no sites there. But you can see we will and do have a fair amount of information in terms of addressing limiting factors. Same thing here in the Little Humboldt. We didn't finish the self-work Little Humboldt complex this year, but there is a fair amount of information. We're going to finish that this year. So we feel like we'll have a fair amount of information again to understand is habitat and where is habitat limiting LCT populations. So just going to provide you some results. These are not accounting for that landscape context that I provided earlier. These are just summaries of data, right? That's going to be something we're going to do next year once we have a little broader inference going to different management units. So one of the first things that we are really interested in thinking about is how connected these streams are to the floodplain. So on y-axis here is vertical disconnection. This is the height, the vertical disconnection between the height of the Bankful channel, the height of Bankful, and the height of the historic valley bottom. And on the y is different populations. Left panels on all these will be the Upper Humboldt. The right panels on all will be the Little Humboldt. A couple things that are worth noting. A lot of these streams are disconnected from the historical valley bottom, right? What is driving that? And is it attainable to go back to historical valley bottom? Love to answer those questions, but I'm not going to cover right this second. Second, within populations, we see a lot of variation in terms of how disconnected or connected these streams are to their valley bottom. Highlighting again, the challenge of using, if you've seen me present up here before, the challenge of using reach-based site-specific methods here, and then I'll go 5 miles away and sample again. The variation, we're not going to capture it. So anywhere we can try and gain some continuous measures of things like connection to the floodplain that we know is super important is something we want to do. And Wally's going to talk about how we've been addressing some of that, as well as Ken. Third, you do see, again, a fair amount of disconnection across populations, but some really stand out. I'm not picking on any populations or habitat or anything, but if you look at like Jackstone and Sherman, those are both small tributaries, nearly 2 meters disconnected between the top of the Bankful and the valley bottom, right? So these are streams that are down. That means water's down. That means less chance for storage, all those things that we know are going to be increasingly important. Same sort of thing in the Little Humboldt. Again, we'll provide more on this in the next couple of talks. And you see this variation across some of the streams. The two figures on the left are from Hanks Creek. The upper Hanks is shown, I think that's above the confluence with the North Fork, South Fork. And you can see it's not very disconnected. You see some Silver Sage in the floodplain, not a bad looking area. I'm not saying the bottom is bad either, the bottom picture of Hanks, but quite a different system, quite a different extent in terms of vertical disconnection to that floodplain, right? So again, we're seeing possible problems, but we also see the need to sample extensively across these areas. And then others is Jack Creek tributary to Maggie. That's, I think our field crew estimated about 3 meters disconnected between the stream and the historic valley bottom. So some of these are not necessarily what we would think of as ideal habitat, but more to come. Second, we know an important thing we've been, if y'all remember any of your geomorph classes or any of your fish habitat relationships, just how important woody debris is, right? And so here on the top, I have large woody debris on the Y, populations again on the X, and on the bottom is small woody debris. So small is just less than 10 centimeters in diameter, 10 centimeters in diameter. And a couple things you can see here across almost all the populations, there's very little large woody debris in these systems. It's not surprising, right? This is sage country. God, Robert, you're really doing a good job. But at the same time, what this suggests is we don't really have a lot of that large material that can provide slowing down or aggregation of these streams back up, right? We don't have access to large material that can have large woody debris jams, et cetera, in some of these systems. The good news is there is a fair amount of small woody debris. And that's, if you can imagine, there's a lot of that, like riparian willows. They're not more than a couple inches in diameter, but there's a lot of them. And it does vary across systems for sure. Again, you see some. I'm not picking on Jackstone and Sherman, but Jackstone and Sherman, again, there's not much going on in a place like Jackstone and Sherman, right? So we're seeing some discrepancy across populations, even in the things that could possibly help restore them. This also highlights the importance of structure other than what's on the landscape and thinking about BDAs. And I know there's some projects planned in terms of BDAs, also the importance of beaver. And so you can see this is from dam in the Marys. Again, this is a fairly big river right here, not a lot of large material here, right? So to think these systems are going to do it themselves is Disneyland. Here, we're also accounting for things like jams and so woody debris jams or dams from current or past beaver. And you do see some amount of jams and what we call jams. And again, I work with these guys at Utah State and they've thought about this stuff for a long time, but really is anything that is causing some change to the channel, right? So when there's high water, is that water interacting with something that's going to change the channel or the sediment sorting, things like that, right? So it doesn't have to be like, hey, if you're more than 200 pieces of woody debris, you're a jam. That's silly, right? Because you could have just a couple of pieces that are clearly having an impact on the channel. So when you look at these results, there are moderate amounts of jams. It's nothing that's, again, going to indicate that these systems can possibly do this on their own unless long time periods. Beaver dams, a lot of fluctuations, and we see a lot of like abandoned beaver dams. And what's going on with that is, some people think it's disease, others, it depends who I talk to. I talked to a bunch of landowners. Some people think there's excessive trapping, shooting beavers, et cetera. But we do see a fair amount of abandoned beaver dams on the system. And there are some really good beaver dams, obviously, in places like Maggie Creek or Beaver Creek and some of these other places. The other thing we're seeing is kind of an overwhelming amount of fine sediment across populations. So same x-axis, the Y here is the percent of the pool habitat with substrate less than 2 millimeters. And the red dashed line is from a summary paper that considered the levels that were detrimental to cutthroat trout. And that's not just from LCT, from other populations. And you can see, doesn't really matter what population you're looking at, there's a lot of fine sediment. Some populations, every site we go to, every stream, every pool exceeds that level, right? And you all have walked around these streams. This is not magic, right? If you're shin deep in mud, is that good trout spawning habitat or ability to access interstitial spaces? Probably not, right? We do see some places, obviously, that have really nice sediment quality. This is Upper Tea Creek in the wilderness. nice looking system, but we also see others, and you can see, this is, an area on Hanks Creek, fair amount of fine sediment. Yeah, it's just there, it is just what it is. There's a lot of sediment in these systems. The good news about some of the fine sediment is it does mean there is some sediment available for that aggradation of some of these streams back up, right? So, hey, let's think positively. We also see similar results when we've assembled JAWS data. So this is the same management units or subunits. On the Y here is just 3 millimeters because JAWS, I just talked to Jacob, he said 1/8 of an inch, and that's 3.25 millimeters. So it's a little bit larger in terms of the cutoff, but you see the exact same pattern from the JAWS data. So a lot more data here, adding into ours, same pattern. And when we add in AIMS data, so AIM I just have for by management subunit, Little Humboldt on the left, Upper Humboldt on the right. On the right panel here, same thing, finds less than 2 millimeters, excessive levels. So sediment appears to be a fair problem. Pool habitat, just wanted to show this. There isn't excessive levels of pool habitat, but I was talking with Sherma about this yesterday. Like, people get so hung up on like, oh, we should have 50-50 pools. Like, where the hell does that come from? Depends what kind of stream you're in, right? If you're in a stream that's steep like this, do you expect 100% pools or 50% pools? And then, you see publications that talk about, increasing pool habitat is good for cutthroat trout. One just came out actually. So does that mean if we had 100% of a site was a pool, that's a trough, right? Or a slough? Is that good habitat? Probably not, right? So this is a metric that really has to take into account what's going on in the context of the landscape. But in general, there are some places where you see very few amounts of pool habitat. Take that for what you'd like. Again, pool volume. This is residual pool depth. So this is from the tail to the deepest point. It's kind of an index of how much volume there is of a pool in terms of like deepness. So if you drained it like a bathtub, how much is left? We know that happens in a lot of these streams and a lot of years like last year, right? Where there's fish just clinging on to that little bit of water. And so I have residual pool depth on the Y, same panels. And you can see really shallow pools is pretty common, with a lot of the tributaries having less than 0.2 meters. So, less than 10 inches of max depth to tail, right? Doesn't bode that well in terms of drought years, in terms of like persisting through drought years. And when you look at this picture here on the right, it's not surprising. These things are filled in with sediment. I mean, we would be calling this a pool and it's filled in with sediment. I mean, it's not surprising that they're shallow, right? So it's linking the sediment, lack of woody debris. There's lots of things going on here that are tying together. Preliminary conclusions. And again, you can read all this in our report. We're not going to send out some exhaustive, like 500-page report. We're trying to provide a condensed or a denser version that people hopefully will read and not just like circular file it. But some preliminary conclusions. We're seeing disconnection from floodplain. It's variable, but extensive. Low amounts of large woody debris, but a lot of small woody debris. Again, suggesting that thinking these streams are going to recover on their own, passive restoration in a lot of places. Maybe not. There's relatively few shallow pools. Again, a challenging thing in terms of thinking about how populations and fish can get through drought periods if the pools are consistently shallow because they're filled in with excessive levels of fine sediment. And the levels of fine sediment, I mean, there's so much work on the importance of sediment, not just for spawning, but access to interstitial spaces to avoid predation, to detecting predators, non-native predators. There's some nice work showing how young year can detect predators in the water, but fine sediment kind of inhibits that. So lots of problems with fine sediment. And of course that has ecosystem effects in terms of food production, things like that. If you have any questions on this stuff, you can ask me, but we have a couple more talks that I think are going to tie this together. And so if anybody has any questions, I can answer them.
Yeah, I noticed in the Maggie Creek Basin, we didn't have endowed JAWS data, but the BLM has very similar data. basically the same protocol as JAWS. And I didn't see that data labeled, and so I think you've got a rich data source there that you could add in.
Yeah, you know, so we have these Habitat Core team meetings, and I just wish someone would tell us that, right? At the meetings. So next year's
I think
report.
I mentioned that.
I more somebody from the, I'm not picking the BLM like Jason back there, or Nick. But sure. Yeah, it's unlikely to make it to this year's annual report, but next year we will. But yeah, those are the reasons that we have the meetings, and hopefully people are forthcoming about those ideas. And so we're not getting them at the end of the year. But yeah, thank you. Yeah, we'll integrate. Any data, like, you know, Summit Lake, we saw that they have a fair amount of data on if you guys have habitat data as we move west, love to integrate it. This is not some, we have to do it our way. We want to see integrate different sources of data and come up with a better assessment.
Another thought is that there are certainly it has been a lot of incision, and so the valley flat that used to be the floodplain is now the terrace, and unless we do massive restoration bringing the channel up, then the way forward typically is by creating and accessing a floodplain inside the gully.
Yes.
And I didn't notice that in the data, and it seems to me that that width of the gully and the floodplain inside the gully would be a critically important thing to focus on, because that's where the large wood in the base of the tall clumped willows comes to live. Yeah, once you get the grazing management right, then you can get the willows, and once you get the willows, then you get the beavers, and then the beaver dam acts like large wood also, and you get pond pools and so forth.
Yeah, well, he's going to talk about some of the proportion that is active, accepting the fact that we can't go to the historic valley bottom. I think that's a decision. If some of the work colleagues from Utah State have done in Eastern Oregon, really in size streams, they've been able to actually grade them back up to those floodplains. And so there's a lot of talk about, hey, is inset floodplain good enough? That's a decision, right? That's not what's possible. So we're trying to look at what's possible, but we will have measures that can get at that. So again, it's a decision saying, hey, we want to continue for land management, whatever anybody's doing down there, right? That's a decision that we don't think we can bring it back up, because we don't know what is possible in most of these places because they've been managed for hundreds of years, right? Arid ecosystem, we don't know what they historically looked like in most places, right? So we're trying to use it as a base, but our metrics will get at that term. I think Wally will cover that.
Robert, there's a note in the chat from David. Is Lower Marys River good habitat for trout migration into the Humboldt River main stem, or are the trout just in the upper tribs as shown on the map?
Certainly just looking at the habitat, is it good for trout migration? In years like, it depends what time of year you're looking, and you know, I'll defer to Jacob and Cody and Mike, but you know, like last year, there's no water in some parts of it in the summer. So if that's your assessment, no. But in terms like of years like this year, then we're going to do some work. I think Wally will talk a little bit about it, but trying to understand what these rivers look like in big water years. So big water years like this are probably going to provide access to the Humboldt and loss of tributaries that aren't commonly connected.
Any other questions from folks online? Sorry, Robert.
No worries.
I don't see anything.
Okay, our next speaker is thanks.
This one.
Yeah, our next speaker is Cash Rasmussen, and he's from Utah State University. He works with Wally in their lab and Nick and Joe Wheaton. And he's going to talk about just how we're using drone surveys to try and gain a broader inference again than these reach-based efforts.
All right. Thanks for having me. Like Robert said, my name is Cash Rasmussen. I'm a GIS technician with Utah State. Today, I'm just going to give a brief presentation on how we've been utilizing drones for the assessment of riverscape health. And as Robert touched on, it's going to be the components of the riverscape that are driving the LCT habitat conditions.
Just click on the screen.
Okay. So when we're assessing riverscapes, we want to be able to sample at the valley bottom scale. What I mean by the valley bottom, it's just the low-lying areas adjacent to the river or stream that could plausibly flood under current flow regimes. So in other words, this is the floodplain. And rather than solely focusing on in-stream conditions, we want to broaden our focus to include the entire lateral extent of that floodplain. And we want to be able to do this over larger areas, and that's where drones come in handy. So using drones, we're able to significantly increase our longitudinal footprint. A field crew will typically sample 150 or 300 meters, dependent on bankful width. Up here, you see a typical drone flight, and this covers about 1.5 kilometers. So anywhere from 5 to 10 times that of the typical field crew. And the main point here is that this allows us to increase our area of inference. So for example, if we look towards the bottom of the screen here, highlighted in yellow, this is a 300-meter reach. If a field crew was out sampling this area, the conditions would vary widely as if they were to, compared to if they were to sample this 300-meter reach just a few hundred meters upstream. So down below, you have a single channel, not much vegetation, not much complexity. And then you go up a bit and you have a beaver impacted site with a lot more complexity. So the point here being that we're able to increase the area of inference and capture more variability within sites. And hopefully this can help streamline field sampling. So if we zoom in on that more complex 300-meter reach, a field crew may have a hard time navigating this. There's beaver ponds that might be too deep to wade through. You have thick vegetation that makes things difficult to navigate through. And especially when you're trying to sample, again, not just in-stream conditions, but the entire extent of the floodplain. And overall, this can just be difficult to capture all the complexity within a site. So there's things like multiple channels that when you're in the field, it can be hard to grasp all of this. And we hope that drones can help streamline that. There's also a time and budget component. So if we zoom back out to this 1.5 kilometer reach, just based on how much time it was taking a field crew, this could take anywhere from 10 to 15 hours for a crew of 2 to 4 to sample, which equates to about 20 to 60 paid hours. And then a single trained technician in the lab, once we have our imagery, could map a site like this in anywhere from 8 to 10 hours. And that translates to about 8 to 10 hours paid. So from June 2021 to September 2022, we flew a total of 81 sites. For each of those 81 sites, we flew about 1.5 kilometers per site. These flights were conducted within the Upper Humboldt and the Little Humboldt management units. So we have currently 121.5 kilometers of riverscape that's been flown. The figure up here is just for the Upper Humboldt. This is where the majority of our flights took place, but keep in mind that the Little Humboldt also has some flights. So the yellow polygons with the red outline, each of those represents a flight path. And then for all of that coverage, we have some drone products. The first of which we've been seeing some examples throughout this presentation. This is our ortho imagery, just a mosaic of individual images captured by the drone. And we export this at about 5 centimeter resolution per site. One of our drones also has a near infrared and red edge sensor. So we're able to calculate things like normalized difference vegetation indices. Ken McGuire is going to go into more detail about how we can utilize NDVI values. But overall, this just gives us pretty good context of vegetation and vegetation health within sites. And we also have digital terrain models, or DTMs. This is produced from unclassified point cloud data that comes from the drone. Unfortunately, it does not bear earth, so it does include things like vegetation. but it still gives us some pretty good elevational context within sites. So in this example, you can see those blues, those dark blues are the lowest lying areas. The valley bottom confining margins are being exposed pretty well. So some useful products. And then we'll use these products to conduct our mapping. So these are all manually delineated features that we map for each site. First being landscape features. These are features that don't really change over time, at least not within the temporal scale that we're concerned with. So for us, this is our valley bottom and valley bottom center line. As I mentioned, the valley bottom is just that low-lying, plausibly floodable area. And then for each of these, we delineate a center line, and this can be used to measure things like sinuosity throughout the site. We also map geomorphic and hydrologic features. So one of those is inundation extent. This is the wetted extent at the time that the imagery is taken. This can be a really good indicator of that lateral connectivity within sites. We classify these as free flowing, typically lacking structure, or structurally forced overflow in the form of secondary channels and sheet flow, and then ponded, so like a beaver pond, anything, a structurally forced pond, essentially. We also map active channel. This is typically found within the bankful width of the channel and reworked by average flows. Channel segments, just another way to map complexity within a site. How many secondary channels are there in relation to the primary channel? Then we move on to structural components. So we map natural beaver dams. We do this in the form of mapping out the dam crest. Up here, you can see some examples of some intact dams. We also classify these by structural integrity. So is it intact? And if it's damaged, to what extent is it completely blown out, or is it just breached? Then we map some other structures to show structural complexity. So if we can see it, we'll map large woody debris, jams, inorganics, things like boulders, and then live vegetation that might be present within the stream as well. And we also map vegetation extents. So we do this in the form of riparian and upland vegetation. So in the green here, you can see our riparian extent, and the gold orange is the upland. We can compare this to things we're finding through NDVI. And then this is also a good indicator of sources of large woody debris into the system. So keep in mind all the stuff I just went over. So these are mapped for each site, and it's just a snapshot in time. What drones allow us to do is to easily capture repeat photos over these sites. And that can be useful in tracking change over time, specifically when it comes to pre and post restoration. So if we want to see how an area responds to a restoration approach, that's known as effectiveness monitoring. And an opportunity we have is, I'm sure many of you are familiar with the wildcat fire within the Upper Humboldt, broke out in July of 2022. We were able to capture some pre-imagery of some of the areas that were affected by the fire, as well as some post. So here is just these two images alone can give us a pretty good indication of how these sites were affected by the fire directly. And then in working with the BLM, who's secured some emergency funding for a restoration approach, we hope to be able to continue flying these sites and see how they respond. And this is a site on Tea Creek, I believe. So I've been up here just blabbing on about how great drones can be. However, I did want to highlight some of the limitations. So flying and data capture. So when you're in the field actually collecting this data, there's some things that you need to consider. Batteries and charging. So each drone is limited to about 20 to 30 minutes, depending on the drone, per battery. So when you're flying as much as we did this last season, it's important to have enough batteries and a reliable way to charge those batteries while in the field. Weather, if it's raining, if it's too windy, you can't fly. So it's really important to pick nice weather windows to collect this data. Spatial limitations, we discussed how the drone can be used to address the finite sampling window of field crews, but we're still spatially limited in the sense that we can't yet fly an entire Huckgate watershed or an entire management unit. So that's something to consider. And we still need those coarser but widely available data sets. And then technical difficulties, user error. Unfortunately, I can relate to this guy. Last year, flying, everything was going fine. And then in a heartbreaking turn of events, there was a connection error with the drone and it was never seen again. But we had an extra, a spare drone. So we were able to continue flying with relative ease, but just things to consider. And then once we have all this drone data and we're ready to map, we were hoping the drone could do everything, but of course it can't. So one of those limitations is of course substrate. In the field, they're collecting substrate to a pretty fine scale. And we just can't really get to that scale with the drone products. Vegetation obstructions, simply put, if you can't see it, you can't map it. So anything that's going on within the stream, if there's too many vegetation obstructions, then we just can't really do much with that. And then vertical aspects. So things like incision are very difficult to map from aerial imagery, even high resolution aerial imagery. Even with, I pointed out these DTMs, here's a DTM for Current Creek in a more confined system. Notice here, you see all of this texture within the channel. That's all that vegetation that's being represented. So we just can't do much in terms of mapping what's going on beneath that vegetation. Not to say it's useless. We can still map things like valley bottom to a high degree of accuracy. From here, the ortho imagery, you can see that vegetation. So we're still able to map riparian vegetation, but just some limitations to consider. So yeah, in conclusion, drones allow us to overcome finite sampling, especially when it comes to field crews. Photo points, these are just photo points on steroids, and we should be utilizing this relatively cheap way to capture repeat photography for sites and track change over time. And overall, we think that drones can be a powerful tool when used in conjunction with field sampling and coarser data sets. And I did also want to mention that all of the data that we've been producing, we've been working with Matt and Leaf to get that onto the conservation efforts database. So yeah, that's all I got. I don't know if I left much time for questions, but that's what that says in the corner.
We're going to hold questions until the end because we have an hour or so to have a specific discussion around habitat assessment.
Cool. Thank you, guys.
Just 2 questions.
We're gonna do them at the end.
For habitat discussion, so we're saving all that.
Who's next?
Loop it all together with all the speakers.
I'll track the questions in chat, just so you know.
Thank you. Right, I'm Ken McGuire from Desert Research Institute here in Reno, Nevada. And I'm going to be talking at the coarser scale over longer time scales as well of how satellite imagery and geospatial sorts of techniques can provide a value-added way to drive you towards sites that are good candidates and hopefully not sound like a wild man that says the car will drive itself. The concepts that will kind of drive the discussion here are how we can use this type of technology to look at the status of what's there, how the landscape has changed over time that might be useful, and also indications of how resilient the location is as well. So for the riparian status, I've been using mostly normalized difference vegetation index, which was mentioned previously by Cash. It's a measure of infrared and red wavelengths that is correlated to the amount and vigor of vegetation, terrestrial vegetation. And so we're looking with this sort of measurement at the terrestrial vegetation surrounding the actual channel itself. We're not looking at in-channel attributes.
But hopefully we will have a good signal of the availability of water over time as a informative aspect of how to survey a huge area. We're talking about 500 kilometers east-west, 500 kilometers north-south. The satellite approach that can take a snapshot over a large area of time gives us a way to capture that and rapidly give ideas about where the habitat might be more or less beneficial for the species we've selected. So looking at the normalized difference vegetation index, how does that terrestrial measure relate to what the fish might be experiencing? So if there's a higher or lower value of NDVI, it might indicate which areas are bare versus vegetated. It might correlate with the amount of trees and shrub shading, which might go to the water temperature, and also to an indication of what the long-term water availability of that site is over time. And since it's a spatial measure as well, to the degree the spatial resolution of the satellite system can characterize it, we can look at the proportion of valley bottom that might be occupied by an active riparian corridor. So just to give an example of NDVI and what it might give us that we don't already have, if we look at this area of Hanks Creek, where Connors Creek comes in the Marys area, the existing map data indicates that all of Connors Creek in this area is intermittent, not perennial. But if you actually look at imagery of the area, you do actually see indications that there might be a persistent water source in part of that reach. And if we look in that red box I've highlighted and zoom in a bit, we do see on the right side here indications of a water-filled channel. About 2/3 of the way from the right to the left, it peters out though before it gets to that turn. And if we look at NDVI from the Sentinel-2 satellite imagery, we see within that red box that same pattern. You see that very vigorous vegetation signal coming about 2/3 of the way across the box and then peters out before the turn in the creek there. So just a kind of an example of how that measure from the satellite is relating to water availability on site. So we can look across an entire area and characterize, how does the NDVI vary across the area, and use that to help drive our decision making. And what we've done in this approach is we've taken the lateral variability out of the stream channel, and what we're doing is getting aggregate statistics going downstream for segments of the stream that have a length that's proportional to the width of the channel in this area. And to select the area that we're going to extract the statistics from, we're using the Valley Bottom Extraction Tool polygons that are created by the Utah State crew. And those have also been masked out for agricultural and urban areas within that happen to happen within the valley floodplain. So the issue we have here is that we want to get an aggregate measure that is stable over a geographic area, and we want to be comparing apples to apples. And what we saw earlier in the talk was that the upstream areas are more confined and the downstream areas are less confined. And so what we're seeing is actually an artifact in this NDVI where the areas that are downstream tend to have lower values because we're averaging in a lot of valley bottom that's actually not the active riparian area. And on the upstream areas, it's actually, because it is confined, we actually have a more one-to-one relationship between the riparian area and that valley bottom that's been extracted by the Utah State algorithm. So if we look at this particular slide is created by Hallie, and for the three focal watersheds that we've started on, basically what it's a correspondence analysis, and the kind of overlying concept here is the x-axis is the first axis of a correspondence analysis, a sort of principal components analysis, and it explains the majority of the variation of what we're seeing in that NDVI measure. And it really correlates to elevation. It's kind of indicating a problem with the approach we're using in terms of using the full valley bottom as the sampling unit when we're trying to extract our satellite measures. So I'll get back to this in a later part of the talk, but for the moment, what we can do is in terms of exploring this concept of how we aggregate and get to, from data to information, is to what I did is I used the elevation data to just back out the residuals from an elevation relationship in a regression to give a product where we look at the difference of NDVI from what would be an expected value at that elevation. So if we regress NDVI versus elevation and take the difference, this is the residuals. And this is just a first order attempt for the purpose of this presentation to talk about that, moving from what is NDVI to what is actually a meaningful parameter for the fish. And like I said, I'll wrap back around to how we might address that averaging issue in the future part of this talk. So here, after differencing out, what we're getting more at is what's the difference of NDVI from what's expected at elevation. And then that would help us drive to what might be a more less beneficial site. And we want to take that sort of determination, also aggregate it up to a level which is useful for management. So if we choose a management unit, we want to characterize, what is the satellite data telling us of the benefit of this site versus another site at a coarser scale. Here I've overlaid HUC 12 polygons on top of that previous product. And I've taken the NDVI and I've looked at those segments that were sampled within the stream. And I say, how many 300-meter long stream reaches have at least the expected NDVI for that elevation? So they're not depauperate with respect to the expected NDVI at that elevation. And we see that the lower Marys in the cooler tones and down towards Lamoille Valley and Star Valley pop out from that sort of analysis. And the blue is the overlaid network itself from the valley bottom extraction tool. Or, sorry, we might take, page down. Oh, okay, thanks. Or we might be able to link those segments up and say, what is the longest reach of connected segments that meet this criteria within a given HUC 12. And so in this case, the longest stretch is the actual interconnected full bottom of the lower Marys within that HUC 12 that is occupied with that single constraint. So that's just looking at it in terms of like a measure of the status of what's there from that NDVI perspective. But we can also get other variables from the satellite analysis. So again, so we can look at trends over time. So Landsat has been up in orbit since 1985, and we can look at long-term trends from that data set and say, how stable has this environment been, or is it changing in a way that might be improving or actually becoming worse? We can also look at shorter time frames to look at disturbance regimes and recovery from a particular disturbance event. And again, since it's an image-based data, we can look at the spatial stability of what's going on with the trend. Is it maintaining a similar sort of NDVI, but is the stream actually moving over time? So here's an example of pulling out trends from Landsat over time. So on the upper left is a figure that's color-coded for the trend where warm tones have been going down in NDVI over time in this section of the Upper Marys River by Orange Bridge, and blue areas have been actually increasing over time. The top right plot shows the actual progression of NDVI values from on the x-axis from 1985 to 2021. And we can see the values were increasing starting in the early 90s up to a level that kind of stabilized in the 2010 sort of time frame. And looking at the photos in the bottom, historical photography, we see in 1994, before it started really climbing, there was a lot less vegetation in the channel. It looks like a lot of sediment was going through, and there's not much in terms of impounded water. It just seems to just be flushing through a lot of sediment. And then by 2014 on the right, we have the establishment of more permanent vegetation. In the bottom right corner of that color image, we see some beaver ponds that are established. So we have this ability to take the historical context of what's going on in the site into consideration in our decision making about whether or not we consider a particular site to be more or less beneficial. Now, I mentioned one of the things we want to look at is also resilience. In this particular context, we're thinking about this in terms of drought sensitivity. Looking forward, we might also infer that if something is sensitive to drought now, it might be more sensitive to climate change going forward. Though that is a step logically beyond it. If we are looking at the sensitivity of drought, we're looking at how persistent the water flow is. And also if the water flow is not consistent year to year to year, there also might be a lot of variability in the stream temperature. And we can correlate the NDVI measure with measures of drought like Palmer Drought Severity Index or the Standardized Precipitation Evapotranspiration Index, and use that as an indicator of, is this particular site showing a lot of drought sensitivity? So for this example, the color-coded line on the left is it's indicating the degree of correlation between NDVI and the Palmer Drought Severity Index along the stretch of the stream on Draw Creek. And the cool blue colors say there's low correlation, and the warm colors show high correlation. So the blue site at the top here that's got the photograph with the nice verdant beaver pond area really pops out. And if we look at the time series on the upper right, the blue line shows that it actually has increased a bit since 1985 to about 2000. It's been really stable since, and it really doesn't show a lot of interannual variability. Whereas if we go down to the bottom image that comes from that warm color area, this is part of Draw Creek that got captured by a road that went through, and it's been down cutting, and it's probably dewatering that whole meadow area. And now if you look at the time series, the dotted black line is the Palmer Drought Severity Index, and you can see how very closely the NDVI for that site follows the Palmer Drought Severity Index. So this gives us an idea about suitability for habitat. So we can combine these various measures. So here let's say, how many of those 300-meter segments do we have that has an NDVI that's at least typical for that elevation, that has no significant negative trend since 2013 in the recent past, the last 10 years, and also has a correlation to drought status that's lower than the average in the entire area. So it's not very droughty. And we see in this case that in the gray, a number of the sites don't actually now meet those multiple criteria. And we also see a real focusing in on actually the Star Valley areas potentially meeting these multiple criteria. So we want to move from these raw measures like NDVI to a statement of what is the threshold that's an acceptable thing from multiple criteria, and then aggregate to these decision-making units for where we would focus, where we might send in the drone team, for example, to get higher resolution info that you can't get from the satellite or a filter. This is that measure again of what's the maximum segment length that reaches these multiple criteria. So instead of being all of Lower Marys, when we consider all these criteria being 29 straight kilometers, meaning that when we do these multiple criteria, we actually see in Star Valley, we get a 6-kilometer connected stretch that meet all those multiple criteria. We can also then aggregate up to large areas if we want to look at like one GMU versus another and say how is the habitat status in this one versus that one. We can look at the histogram of values, and here on the left we have the Reese River sites that we've been doing in the past years, and versus the Upper Humboldt on the right. The two top ones are the number of segments that would exist that are 300 meters long and uninterrupted. The bottom is the maximum segment length that exists with those criteria. The blue is if you just use that one single criteria about NDVI above the expected, and the orange is if you actually have the multiple criteria. And what we kind of see is that the number of segments and the lengths are so much longer in the Humboldt than in the Reese. But when you actually put these multiple criteria down, the Reese actually maintains the distribution of orange and blue values is less strongly affected than in the Humboldt. In the Humboldt, when we have these multiple constraints, a lot of the areas drop out proportionally speaking compared to the reefs. So getting back this, what I've been doing here has been just doing this simple adjustment for elevation to try and compensate for the change in confinement and trying to make it an apples comparison between the bottom parts of the river network and the upper parts that are more confined. What I'd like to move towards is actually finding the inset part within the valley bottom that is the actual part that is the active riparian corridor, so that our statistics are not being diluted by upland areas that are being averaged in, because when it's unconfined, often these rivers don't fill the whole valley. Here on the Marys at the top, you see actually the river does fill the valley bottom, but as we move down on the left image, we get proportionally less of the actual sampling area that we're averaging being the actually riparian area. So what we might do is we might have a threshold NDVI and say, let's within that valley bottom segment out what seems to be active riparian corridor. The challenge though is that on the right side of that image, you'll see there's a snaking down channel that appears to be disconnected. And this is going to be something that I think Wally will get at too, is this idea of maintaining information about what is connected hydrologically to the channel. But what I'm thinking about here is that the distribution of NDVI values within this, in each of these sampling segments might be used. So you see, even though on the right image, if I have a threshold value of NDVI that seems to pull out the riparian vegetation, it gets both this disconnected part of the a channel that's relict or due to an avulsion, it's not there anymore. But on the left-hand side, the green color is showing that there are values that are very high NDVI. So you'll notice this, if we go through these segments, you'll see that maybe there's a way we could use the high values within a connected area to identify the most likely channel location that's hydrologically connected, and then clip out the blue areas that don't have these very high areas. So look using the distribution of NDVI values instead of just a threshold as a way to try and get at the active riparian corridor. So from here, we'd want to, once we're happy with what we've done, we'd want to bring in information from FlowPer, about flow permanence. We'd want to bring in any modeling of stream temperature to create those sorts of graphics I was giving about, what candidate areas or what proportion of the area might need a desired sort of environmental state. And then that would be used to drive decision-making about where to send people in the field to get the sort of information that we can't get from this sort of data, like sediments and the amount of woody debris. So thanks to Fish and Wildlife for the CESU support to do this, and to the rest of the Habitat team, Robert and Haley and Wally and the Utah crew like Cash.
People feel free to stand up a little bit, move jobs around a little bit. This can be tough, like right before lunch, this last comment.
All right. All right. Thanks everybody for your attendance. And this has just been an awesome opportunity for me to be here. I've learned a ton about genetics, and my brain is like fried right now, so I'm going to try to make it through this. But I wanted to thank my co-authors that are listed here, and I'm going to dig in a little more about just floodplain connectivity and maybe go into some concepts and show some pictures. So this is a picture of an area that's well connected. So you see sheet flow across here. We see this opportunity when these are laterally connected. There's frequent overbank floods. This increases soil moisture. So this idea of the sponge, it's getting moist. It's facilitating seed dispersal of microbes and settlement, nutrients, and so forth. And so, as Sherm kind of pointed out too, that floodplain connectivity is determined by that channel morphology. So what's happening as far as incision and how much wood is in the system. So you'd expect that this system, if you went over to the stream channel, the main stream channel, you'd probably see little to no incision. You'd probably see a lot of large woody debris or some sort of beaver dam or some type of activity. So this is kind of like the poster child of what we want to see across the landscape. And so why is this important? So this is a shot of Bridge Creek in Oregon, and this is a beaver dam complex. And it's just kind of maybe ironic that beavers are trying to do exactly what we're trying to do, in that they're trying to connect the floodplain. They're doing it because they feel a lot more comfortable in water. So they're connecting these areas so they can go out and they can forage further. And so the benefits are widespread. So if we look at just the flow, if we look at the in-stream channel here, we look at the hydrology, the hydraulics are diverse. So we have pools, we have backwaters, we have eddies, we have all this hydraulic complexity that wouldn't exist without being connected, without having in-stream wood. And so once again, we have that retention of water, sediment, nutrients. We increase that riparian productivity. And we have that complex habitat we're looking for, and the resiliency. And of course, the benefits that we're looking for Lahontan cutthroat are there in these type of systems. So kind of our working hypothesis is that more floodplain connectivity is better for LCT. So we get happy fish when we have well-connected floodplains. And so perhaps floodplain connectivity can be used as a surrogate for a measure for in-stream LCT habitat. That's yet to be determined, but that is one of the hopes, because this is something we can measure from space, as Ken's talking about. We can get at floodplain connectivity from space. We have a lot harder time looking at actual in-stream conditions, just because of the footprint of the whole floodplain compared to the in-channel. So we've got examples of where we have high floodplain connectivity, like this is the Lower Marys, and there is good intact stream habitat in those systems. So say in the Lower Marys where it's well connected, if we look at the inset photo there, we see that we have pools, we have riffles, we have complex habitat as well. And then in a degraded system, this is Lower Current Creek, kind of the opposite. This is an area that's pretty much disconnected from its floodplain, and it's pretty limited, the in-stream habitat. Maybe you just have kind of run and not much complexity as far as habitat. So kind of the question is, we see these on the landscape, especially in these extreme cases, but how do we get at more of the nuance in across the whole range? And so incision is typically one of these causes of disconnection, and Sherm brought it up. These are widespread throughout the Great Basin and in the Lahontan Basin as well. So the idea here is, on the top slide, we have an intact riverscape with the green. We have this well-connected floodplain. We have multiple channels, and then below we have where this stream is down cut, and now we have the orange is kind of taken over that area. And so, and then down here, Kevin, we've lost that riparian. So we've really constricted the riparian. Here's an example of Draw Creek, just to kind of emphasize that. So I'm going to talk about this idea of how do we get at lateral connectivity? How do we measure it? Floodplain connectivity. And there's multiple lines of evidence that we've approached, and we've talked about a lot of these already. And so some of this will just be kind of pulling it all together and seeing how it works together, kind of in concert. So first of all, we've got Ken McGuire's done a great job of mapping riparian vegetation throughout the state of Nevada. He has a product called Marvin. It's map of riparian veg for Nevada. And that's something we're using, as well as, flying drones to map the riparian vegetation. So this would be the Marvin product, just binary. So the green would be riparian areas throughout the Upper Humboldt Management Unit. Ken talked about NDVI, where, satellite based, and then Cash talked about it, drone based NDVI values. And so this would be just a simple product that Ken developed in NDVI, kind of this late summer mean averages. So these, the reds would be low NDVI, and then the green values would be higher NDVI. And these aren't normalized by elevation like he had suggested, but just to give you an idea how these kind of work together, that'd be another product. And then in the field, we can actually try to map more of the active floodplain. So as I just described earlier, a lot of these riverscapes have been down cut, and now they have a component of the floodplain that's just not being flooded very often. So it's inactive. So we can go out in the field, and with the GPS unit, we can go out and measure those locations, get an idea of, okay, this is our active component. And the nice thing about having that field data is you can come back in and you can use that to train other remote sense data. So for example, one thing we're doing to get at incision, to get at this vertical component of like why we've lost some of the floodplains, is we're using topographic evidence for active valley bottom. And so if we look at this slide here, this is based on a 10-meter DEM, and these are based on slope and height to nearest drainage. So the idea of the green colors here, the greens and blues are low elevation, where you can break out, first you can break out valley bottoms or valley flat with that information. But more importantly to this, you can also take that finest component, the very lowest, and you can come up with what you might be pulling out as active valley bottom. And this can be calibrated based on that field data. So you can basically slide it up and down until it kind of matches your field data. And this would be a representation of that. And across the landscape in blue here, we can look at what that looks like across the Upper Humboldt management unit. And we can see that within the valley bottom, a lot of times it's a very limited component, that actual area that can be flooded, right now, that's actively part of the valley bottom. And if we do it proportionally, so this is proportionally across the landscape. So our oranges are a very low proportion of the landscape that is connected. And then as we get into the whites and purples, we can see areas that are higher, have higher connectivity. And so we see the variation throughout the landscape. And so, from kind of measures like this, the hope is that we can kind of predict where healthier habitat might be. So another thing we can do, so sometimes our riverscapes are inaccessible because of anthropogenic margins, or maybe better stated, levees, dikes, roads, and so forth. So in this slide here, I'm showing inaccessible areas versus accessible. So red areas would be areas that are just not accessible because of anthropogenic constraints. And that can be used in conjunction with our topographic evidence. So in some cases, just because something could be floodable because it's low enough, it's not floodable because of anthropogenic inputs. And so that becomes another important line of evidence as we try to really tease out what's active and not active. And then kind of the gold standard for mapping this stuff is actually going out and taking advantage, I think in this case, of we're having just an incredible snowpack right now, over 150% of normal throughout the Lahontan Basin. And we really want to take advantage of this next spring and go out and actually do the inundation mapping. So as Cash talked about, this idea that you can fly your drone and then you can map that wetted width of what type of inundation you see, whether it's free flowing, overbank, or ponded. So why so many lines of evidence? Why are we doing so much work? I think initially, and maybe this is my naivety, and maybe with a gray beard, I should know better, but I thought maybe, NDVI, we're going to be able to just kind of map this stuff and be able to break out riparian versus non-riparian. But there's a lot of complications within the Great Basin. And one of those, kind of the elephant in the room, in my opinion, is these relic willow stands that are up on these inactive surfaces, and they're surrounded by upland. And so they really throw off the NDVI values. And so a lot of what we're trying to get at is to be able to tease out these species. And I think that's where those other lines of evidence are becoming really important. And so kind of where we're at right now, what we're doing right now is we're trying to bring it all together. It's similar like listening to, you know, the genetic stuff. Is like we're learning as we go. And in the case, we're trying what we've learned in other areas, but there's a lot to learn in these systems. And we're trying to compile it all and bring it all together and, you know, come up with good indicators of riverscape health. One thing that I just wanted to show in that regard. So this is a map just showing NDVI and proportion of active valley bottom. And this is a bivariate map. So it's basically NDVI and active valley bottom that was derived from the topographic map. So the idea is like the grays on here would be low NDVI and limited active valley bottom. And so the white to gray areas are what could be considered, you know, potentially as poor habitat. And then the dark greens would be, you know, the better habitat. And so these are things that we're going to kind of attempt to follow, you know, see how these kind of tease out and see how they work. But the relationship looks pretty good. I mean, so we see, so the y-axis we have NDVI, and then in the x-axis we have proportion active using that threshold from the field data. And so you see we see a pretty positive relationship. The error bars are giant, and we do see a dip down as things become really connected. So that final bin on the end here would be areas where it's kind of dropped, NDVI values have dropped. But what we're noticing in those cases, there might be things such as water. This is one of the reaches, and it's all water. So in this case, it might be a well-connected valley bottom, but as we know, NDVI values for water are very low. And so, you know, those relationships are going to have to be really thought out as far as what they really mean. But it's kind of a promising direction. So where are we going to kind of take this and how are we going to use it? So kind of some primary uses of this, in my opinion, at least, is we can come up and identify the degree of disconnection throughout the whole entire LCT domain. So we can have a map of like the floodplain condition, and this will help establish, you know, the status assessment for habitat, and then also the recovery potential, and help with monitoring, like coming up with monitoring, prioritization and stratification, and then help with the restoration plans throughout the basin. And so I'm done with my talk.
I think the plan is to break for lunch and then come back to questions and additional discussion. Is that still where we're at, Cody?
So yeah, we have the entire Habitat Core team here in person on it. Robert has some kind of a brief summary to pull everything back together that we'll come back with after the lunch break on here. So I encourage everybody that is going to be joining that discussion, you know, virtually here, just throw your messages in the chat. We'll get to that, and then we'll do a similar group discussion in the room here after the lunch break on it. Before we do break for lunch, though, wanted to check in with everybody real quick. If you are a RIT lead, or if you were an interested party that was planning on participating in the short-term action plan discussion workshop, if you still want to participate in that or see value in that, please either let me or Rachel Humans know in the lunch break, or put it in the chat here, and we'll kind of gauge some feedback. If it's not going to work for a lot of people with travel needs and stuff on it, we can also pull together a virtual workshop sometime in the next week or two. So we will see everybody back at 1 o'clock.
Thanks, Cody.